The next generation of the nation is born, grows, and is ready to serve public health, a place where knowledge, innovation, and dedication come together as one. Welcome to the College of Pharmaceutical Sciences, Yayasan Farmasi Semarang, an institution of higher education committed to producing professional and excellent pharmaceutical personnel in Indonesia. Located in the city of Semarang, this campus is the best choice for young generations who want to pursue a career in pharmacy. With modern facilities, a comfortable environment, and experienced educators, we are here to provide quality education oriented towards the development of pharmaceutical science and technology. Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang is supported by complete and advanced laboratories. Various modern equipment is available to support student practicums, ensuring they gain hands-on experience in the real world of pharmacy. We believe that maximum practical experience is key to producing graduates ready to compete in the workforce. With a research and technology-based learning method, our students are equipped with the skills needed in the current pharmaceutical industry. Supported by an innovative education system and competent educators, graduates of the School of Pharmacy, Yayasan Farmasi Semarang, have made their mark in various fields. As part of the 3 pillars of higher education, we are active in community service activities, ranging from health education, social service, to pharmaceutical services for the broader community. This is concrete evidence that the knowledge we provide is beneficial not only for students, but also for the community. Not only academic knowledge, we also offer soft skills training to support students in facing global challenges.
Assalamualaikum warahmatullahi wabarakatuh. Kami dari Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang, biasa disingkat dengan STIFAR Semarang. Sekolah ini mempunyai 5 program studi berurutan dari awal lahirnya adalah Diploma 3 Analisis Farmasi dan Makanan, Diploma 3 Farmasi, Strata 1 Farmasi, program studi pendidikan profesi apoteker, dan yang terakhir adalah program magister farmasi. Kampus ini terletak di Jalan Lejen Sarwo Edi Wibowo, Pucang Gading, Semarang. Para lulusan kami sudah tersebar di berbagai kota di seluruh Indonesia, dan mereka telah mendharmabaktikan, mengaplikasikan ilmunya pada berbagai bidang pekerjaan, baik itu sebagai pegawai negeri, pada bidang pelayanan di rumah sakit maupun di komunitas, juga di pemerintahan, Dinas Kesehatan, Balai Besar POM, Badan POM, juga menjadi dosen di beberapa perguruan tinggi farmasi, baik negeri maupun swasta. SDM atau dosen yang ada di STIFAR Semarang terdiri dari mereka lulusan strata 3 atau doktor dari dalam dan luar negeri, dan minimal dosen berpendidikan pasca sarjana. Kami senantiasa berinovasi dan selalu berusaha untuk maju dan memperbaiki diri agar menjadi institusi yang terkemuka di negeri ini. Wassalamualaikum warahmatullahi wabarakatuh.
STIFAR Life, lifelong innovation for excellence.
Good morning, everyone. We are about to begin the international conference. Before we start, we kindly remind everyone to turn on the cameras during the conference.
This is where the next generation of the nation is born, grows, and is ready to serve public health, a place where knowledge, innovation, and dedication come together as one. Welcome to the College of Pharmaceutical Sciences, Yayasan Farmasi Semarang, an institution of higher education committed to producing professional and excellent pharmaceutical personnel in Indonesia. Located in the city of Semarang, this campus is the best choice for young generations who want to pursue a career in pharmacy. With modern facilities, a comfortable environment, and experienced educators, we are here to provide quality education oriented towards the development of pharmaceutical science and technology. Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang is supported by complete and advanced laboratories. Various modern equipment is available to support student practicums, ensuring they gain hands-on experience in the real world of pharmacy. We believe that maximum practical experience is key to producing graduates ready to compete in the workforce. With a research and technology-based learning method, our students are equipped with the skills needed in the current pharmaceutical industry. Supported by an innovative education system and competent educators, graduates of the School of Pharmacy, Yayasan Farmasi Semarang, have made their mark in various fields. As part of the 3 pillars of higher education, we are active in community service activities, ranging from health education, social service, to pharmaceutical services for the broader community. This is concrete evidence that the knowledge we provide is beneficial not only for
Are
students.
you ready to experience the future? A place where innovation meets creativity and ideas turn into impact. Get ready to be inspired, challenged, and amazed. This is SINACON 2026.
Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Good morning, ladies and gentlemen, respected speakers, honored participants, and colleagues from around the world. A very warm welcome to all of you. We are delighted to have you join us today for this prestigious Second STIFAR International Conference 2026. Present your research, exchange innovative ideas, and contribute to advancing sustainable pharmaceutical and health science in an international academic forum. SINACON 2026, with the theme Pharmaceutical Sustainability, Bridging Green Pharmacy and Health Science, brings together distinguished scholars, researchers, practitioners, and students to discuss emerging developments in green pharmacy, pharmaceutical technology, clinical pharmacy, and sustainable healthcare system. Hi everyone, I'm Salsa Alivia Delkova Zain, and I'm here today with my precious partner. I'm Maishira Gracia Arvina. On behalf of the organizing committee, it is our great pleasure as Master of Ceremony today to welcome you to this academic gathering where knowledge, innovation, and collaboration come together. Before we begin our agenda today, let us express our gratitude to God Almighty for His blessings and guidance, so that we are able to gather here today, virtually but meaningfully. Now, let's proceed to the opening ceremony. We will listen and sing together to the Indonesian National Anthem, Indonesia Raya, followed by the March of Sekolah Tinggi Ilmu Farmasi Yayasan Farmasi Semarang.
Ladies and gentlemen, allow us to read the ground rules of this conference.
All right, so the first rule is fulfill your nickname with format name underscore institution. Second, please turn on your camera and put the virtual background provided by the committee. Next, fill the attendance link provided on the chat room. The attendance link will be posted via online chat room before the break time and closing session. You will have to fulfill 2 times, and this attendance is required by the audience and participants to get the certificate. And then we would like to ask you to mute your microphone during the seminar. You will have the opportunity to engage with the speakers and each other via online chat room. But still, please be respectful of the other participants and speakers. Keep your comments on topic and succinct. Last but not least is participants are encouraged to take notes during the presentation sessions to enhance their understanding and engagement throughout the conference. Submit your questions to the speaker on the chat room, or you can ask it directly during the Q&A session. You may mention the speaker's name to address your question. And that's all the rules that on this conference.
Ladies and gentlemen, we would like to greet our honorable speaker who are joining today's international conference virtually through Zoom.
The invited speakers, the first one is Assistant Professor Faiz Iqbal Khan, PhD from Department of Bioscience and Bioinformatics, Shan Julthong, Liverpool University.
The second is Professor Gan Siu Hua, PhD from Faculty of Pharmacy, University of Cyberjaya, Malaysia.
And the third is Mr. Bayu Trimurti, Sarjana Farmasi, Master of Applied Science, PhD from Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang.
The fourth is Professor Wafa Al-Jamal, PhD from School of Pharmacy, Queen's University, Belfast, Northern Ireland.
Ladies and gentlemen, today's program will consist of opening ceremony, opening remarks, keynote sessions, discussion session, the announcement of the best presenter of oral and poster presentation, and ended with closing.
We kindly invite everyone to take their seats as we prepare to start the opening remarks.
Ladies and gentlemen, next we are honored to invite the chairperson of organizing committee of the second STIFAR International Conference 2026 to deliver the opening remarks.
Ladies and gentlemen, please welcome Mr. Bayu Trimurti, Sarjana Farmasi, Master of Applied Science, PhD.
Assalamualaikum warahmatullahi wabarakatuh. To distinguished keynote speakers, respected colleagues, fellow researchers, students, and participants from across the globe. Good morning, good afternoon, and evening to you all. On behalf of the organizing committee, it's my great honor and pleasure to welcome you to the second STIFAR International Conference of SINACON 2026. Although we are gathered virtually today, the spirit of collaboration and our shared commitment to advancing pharmaceutical sciences transcend the boundaries of our screens. As the chairman of the organizing committee, I'm humbled to see such a diverse group of experts and enthusiasts joining us today to discuss the future of pharmacy and global health. The main theme of our conference, which is pharmaceutical sustainability, bridging green pharmacy and health sciences, reflects a strategic commitment to advancing environmentally responsible pharmaceutical practices while strengthening their integration with health sciences. This focus underscores the growing importance of sustainable approach in ensuring both innovation and long-term public health impact. Indeed, The concept of green pharmacy strongly aligns with Indonesia's national development priorities, particularly in promoting environmentally improving public health outcomes and supporting innovation in science and technology. Furthermore, these efforts are closely connected with the United Nations Sustainable Development Goals, or SDGs, particularly those related to good health and well-being, industry, innovation and infrastructure, responsible consumption and production, and partnership for the goals. Our conference this year is designed to be a melting pot of innovation. We are incredibly fortunate to be joined by a lineup of highly competent speakers who are true pioneers in their respective fields, including Assistant Prof. Vice Iqbal Khan, PhD from Xi'an Jiaotong Liverpool University, China, Professor Gansi Hua, PhD from University of Cyberjaya, Malaysia, and Professor Wafa Al-Jamal, PhD from Queens University, Belfast, Northern Ireland, United Kingdom. The expertise shared today represents the cutting edge research and innovation in green pharmacy, and I encourage every participant to engage, ask questions, as many as possible, and challenge the status quo during our discussion sessions. In addition, the remarkable participation of more than 100 oral and poster presenters and over 1,000 participants demonstrates the growing commitment of academic researchers and practitioners to advancing knowledge and innovation in sustainable pharmacy. The wide range of sub-themes highlight the interdisciplinary collaborations required to address today's complex healthcare challenges. I would like to take a moment to express my deepest gratitude to the organizing committee. Your tireless work behind the scene has made this virtual gathering possible. To our speakers, thank you for your generosity in sharing your time and profound knowledge with us. I also want to thank our sponsors and partners for their unwavering support. In closing, I hope this conference serves as a catalyst for new ideas, new partnership, collaboration opportunities, as well as new inspiration. May this insight gained today contribute significantly to the betterment of pharmaceutical science and ultimately to the health of humanity. Thank you, and I wish you all a very productive and insightful conference. Wassalamualaikum warahmatullahi wabarakatuh.
Next, we would like to invite Dr. Apoteker Sri Haryanti, Magister Science, the head of Institution of Sekolah Tinggi Ilmu Farmasi Yayasan Farmasi Semarang, to deliver the welcome address. Mrs. Haryanti, the time.
Hashtag. Assalamualaikum warahmatullahi wabarakatuh. Good morning, distinguished guests, colleagues, and participants. It is a privilege to welcome our honorable keynote speaker, one the global expert in computational modeling, natural product pharmacy, nanomaterial-based diagnostic, nanomedicine, and pharmaceutical science. Your present elevate the academic standard of this second STIFAR International Conference, SINACON 2026 Forum. I will also like extend my sincere gratitude to the chairperson of the organizing committee and the entire team. Your hard work at orchestrating this international event is truly commendable. To all our participants, thanks you for joining us today to push the boundaries of pharmaceutical innovation, particularly in green pharmacy. As we greet today, I am reminded of the remarkable success of our first SINACON. Back then, we established a strong foundation for dialogue and discovery. Looking back at 2023, we saw how vital it is to bridge the gap between traditional research and modern technology. Today, we continue that legacy. We focus not only on theory, we integrated the science of green pharmacy, the natural science of pharmacognosy, with the precision of nanomedicine and nanobiosensor, and the speed of computational modeling to solve real-world healthcare challenges. Beyond the exchange of knowledge, the core objective of this conference is to build lasting partnership. I urge every participant to use this virtual platform, not just to listen, but not to connect. Our goal is for today's session to spark future research collaboration, joint publication, and cross-border innovation. Let us transform this digital space into a hub of global synergy. I wish you all a productive conference and look forward to the breakthrough that will emerge from your interaction. It is now my privilege to officially declare the second SINACON 2026 open. Thank you very much. I wish you all a successful, engaging, and enlightening conference. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mrs. Haryanti, for the warm welcome and insightful message.
Before we proceed to the keynote session, we would like to take a moment to capture this special occasion.
Ladies and gentlemen, we will now have our virtual group photo. We kindly request all participants to turn on your cameras and ensure your beautiful smile are ready.
Our technical team will assist in capturing several screenshots, so please stay on screen for a few moments.
Thank you so much, everyone, for your wonderful smile and participation.
Ladies and gentlemen, this is the very moment we have been waiting for, the keynote session featuring distinguished experts from around the globe.
Our first speaker is Assistant Professor Faiz Iqbal Khan from the Department of Bioscience and Bioinformatics, Shanjiaotong, Liverpool University, China.
He will present on the subject Sustainable Drug Discovery in the Era of Green Pharmacy, Computational Strategies for Next Generation Therapeutics.
We will have Mrs. Atika Darumas Putri, Sarjana Farmasi, Master of Applied Science, PhD, as the moderator for this first session. Mrs. Atika is the Secretary of Magister Farmasi Program in Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang.
For Mrs. Atika, the time is all yours.
Okay, good morning, everyone. So does anyone hear my voice here? Sounds clear? Okay, yeah. Hi, Dr. Faiz.
Okay, thank you.
Yeah, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullah.
How are you today, Doctor?
Yes, alhamdulillah, alhamdulillah.
Alhamdulillah. Okay. So yeah, before we really begin, I'd like to introduce you all the participants here about our first speaker today. So let me share the brief. So, okay. Now the screen already shared, right? Yeah. Okay. So Dr. Faiz Iqbal Khan is currently serving as an assistant professor in the Department of Biological Science at Jiaotong Liverpool University in China. He holds a PhD in computational chemistry bioinformatics from Durban University of Technology, South Africa. Basically, he was our mentor back then in South Africa during the my master's program. Yeah, so anyone maybe would like to know more about that. So, and Dr. Khan obtained his bachelor's and master's degree in biomedical science and bioinformatics respectively as well. He also earned a postgraduate certificate in teaching and supporting learning in higher education with merit from the University of Liverpool and holds a fellowship FHEA. That's the prestigious professional qualification, yeah, I think, internationally, yep. And then Dr. Han has also completed the 24-hour instructional skills workshop certificate program focusing on lesson design, active learning assessment, and feedback as well. Throughout his career, Dr. Han has conducted research and teaching at esteemed university in South Africa at Rhodes University. That's the very special university in South Africa and a reputable one in world. And South China University of Technology as well, and also in the University of Electronic Science and Technology of China. His main research areas include protein engineering, protein folding, drug design, and protein dynamics. Dr. Han has established wide-ranging collaborations with BRICS countries, including the South Africa as well and China, and has mentored several undergraduate and also postgraduate students. And notably, one of his students received the International Accreditation Top Project Award in 2024 from the Royal Society of Biology. Of course, during his career as well, he has authored over 90 publications. I think after this, it will reach more than 100 publications in international and high impact factor publisher. And also in recognition of his scientific contributions, this is what made me proud of you, Doctor. He was ranked among the top 2% of scientists in global, conducted by the Stanford University in 2022. Yeah. Dr. Han has received and completed several research grants, including those from the private and also government research funds. and also currently holds adjunct faculty appointments at Taylor's University, Malaysia, Data Megi Institute of Higher Education and Research, also as an honorary appointment from the University of Liverpool. And if we check on this profile, this is really impressive one. But in the actually we got your curriculum vitae, doctor, and that's really detailed, and oh my goodness, it's
I
really
think
impressive. Yeah,
I think
alhamdulillah.
I think it's too much, you know. So okay, exactly. Okay, okay,
Okay, okay,
okay.
yeah,
Thank you so much for introducing.
yeah. I think
So
the time is yours. Yeah.
yeah, exactly. Okay.
Okay.
So yeah. So good morning, everyone. So basically, I am bioinformatician, you know. So most of my research is not just computational work, it's collaboration with the wet lab work also. Okay. So we just validate the results, validate the outcome of wet lab. Okay. So today I'm going to deliver talk on how computational chemistry, that is also a kind of pharmaceutical chemistry, and the molecular modeling, bioinformatics, artificial intelligence are transforming, you know, the modern drug discovery. So the thing is, in this talk, I'm just going to highlight what I did, you know, how I use those computational tools, you know, to predict the proper binding poses, you know, to identify the novel drug. So basically, in this one, in this topic of green pharmacy, I'm just going to relate how computational tools is related to green pharmacy, you know. So I'm going to talk about the molecular modeling and drug discovery, high throughput screening, these terms, you know. Okay. So moving on. So how to hide this, the top bar? Okay, yeah, should be okay. Okay, let me just remove this one. Yeah. Okay, thank you. Oh, can you still see my screen? Green
Yeah,
pharmacy
sure.
and okay.
Yeah.
So I'm relating it with the green pharmacy and sustainable therapeutics, right? So this green pharmacy, just a brief introduction, it focuses on environmentally responsible pharmaceutical research and the development by minimizing the chemical waste. This is just a one of the point, you know, how we minimize the chemical waste, you know. So um, moving on. So starting with my own topic, computational strategies in modern drug discovery. So we all know about the molecular docking. It's a very, very simple. What is docking? Docking is like placing a key into the lock, right? So basically the lock is your target, the protein molecule, and this one, the key is a rigid, the drug. Okay, so drug may fit into the docking, but in real condition, it's not like that one, right? So what is the application here? So from the docking study, we just predict how drug bind to the protein. That's all. This is very simple, but it has several limitations. But it's good for a starting point, you know. The limitation is in nature. In nature, protein is not like, you know, in nature, protein is not static. It's always moving. So when you use computational tools to bind the drug into the active pocket of the molecule, you are just presenting one pose, you know, theoretical pose without any molecular motion, right? This is one of the limitation of docking, but it's a good start, right? So from the docking, what you can do, you know, suppose you have a 60,000 molecule, drug molecule, 60,000, and you have to shortlist. This is one of the database we have of traditional Chinese medicine database. It has 60,000 compounds, and so we are selecting from here to target this lipase, you know, Malassezia restrica, this lipase. We have to target this one. The point is we have 60,000 compound. How to do that? There is one term called virtual high throughput screening or high throughput virtual screening. Okay, so what we did, using the simple molecular docking, simple computational tools to identify which drug is binding with high affinity. So from 60,000 compound, we can shortlist probably hundreds of compound, you know, to make our work ease. And then further, we apply several filters like ADME properties, you know, the drug is suitable or not, the drug is carcinogenic or non-carcinogenic. So we apply several filters that maybe we will have time, then we will discuss, right? So after shortlisting, we may select only 10 to 12 compound for wet lab experiment. So this also ease your burden, ease your burden. And from the first slide, I mentioned about the reduce the experimental burden and also minimize the chemical waste, okay, using the computation tools. So this one is high throughput virtual screening. Another one is high throughput virtual screening is just simple, using one target and the database of the drug molecule. And this is one of the one work from our we publish one paper. Another one is there is a possibility that in case of COVID-19, suppose in COVID-19 we have several protein, envelope protein, M protein, membrane protein, N protein, spike protein, we have these structural protein. We want to see which drug can bind to these all four proteins, so it has multiple drugs, and we are going to shortlist only few drugs and multiple targets. In this case, we selected only a structured protein. So we see the consensus compound. What is consensus compound? Consensus compound is a common compound that show binding affinity or inhibitory potential against all the structures of protein. So we found that these are the compound, like phenylalanine, sepharanthine, and other compounds. They bind to all the structured proteins. So this is one of the good idea. Some of the protein you may encounter that have two sites, two sites, and both sites is important for pathway. Suppose you have to inhibit it, so you have to design a drug, one drug that fit into the both pocket. This is also possibilities, of the application of molecular docking. There's another term called, so we discussed about high throughput virtual screening. There's another term called ligand-based virtual screening. What is the ligand-based virtual screening? We already have one drug. We already know this drug. For example, here, PF543. Okay, this is one of the inhibitor against sphingosine kinase protein. So what is this sphingosine kinase? This protein highly expressed in breast cancer patient. Okay, so during breast cancer, this sphingosine kinase is highly expressed. We already know one of the drugs, like PF543, that inhibit this protein. But we are looking for the better drug, So what we have to do, we have to look for ligand-based virtual screening, searching the similar drug, similar structure, just a few modification of side chain, so that the drug is much more better than this one. That is called ligand-based virtual screening. So we selected a drug bank that has 7,000 molecules. We selected by active compound database that is and commercially available and other virtual libraries. And we use this template as a for the ligand-based virtual screening. So from here, we selected 20,000 compound. Then we applied some filters like drug likeness, ADME properties and pains. Further, we shortlisted 621 molecule. Then we further apply ADME to check the toxicity and all. Then we selected finally 55 hits. So see how, the computation tools reduces your burden, reduces your chemical waste. So from these like 100,000s of molecule, we shortlisted 55 hits that is more similar, more, close to PF543 template, already known the drug. So this is called another term ligand-based virtual screening. So maybe we can apply all these tools, like virtual screening, multiple targets, ligand-based virtual screening. These are just part of, extension of molecular docking. But I also told you what is the limitation? The limitation here is the protein molecule is never in a static form. In our body, in our cell, the protein molecule is always moving. It has atomic motions, it is interacting with other proteins, PPI, protein-protein interactions. It is interacting with other molecules, right? Like in our case, serum albumin protein in our blood, it has multiple binding sites. It carry a lot of molecules. Okay, so in nature, the protein are flexible. So this is one of the limitation of molecular docking. So, and also, the shape of the pocket is changing. Some of the protein, in case of lipase, they have open and closed conformation, open and closed conformation. So when the lipase is open, that means the active site is open. It give you, the pocket is open for the drug. When it is closed, this we can consider that this lipase is inactive, right? So this is one of the limitations. So what is the next point? So after the molecular docking, we got molecular dynamic simulations. So it's kind of bringing back, bringing your rigid protein to the life, to the motion. Okay. So see how in this animation, how this is one of the PD1 PPI interaction work. So these two proteins antibody are interacting, they have a PPI a surface for protein-protein interactions. So it will give you the motions. It will calculate all the atomic motions, all the position of atom throughout the simulation. This is one of the tool, molecular dynamics. These are all computational tools. No need to use a lot of chemicals. You shortlist it and do further validation in your lab. Okay. So this MD show how protein and drug interact over the time, like in our real-time system, real-time body. Okay. And it also validate if docking pose is stable or not. Suppose for docking, this is just a computation tool. It will give you the molecular interaction, right? But it will give you whether this drug is in the pocket or going away from the pocket, right? So this is one of the nowadays we have several tools, like online tool I have gone through. There are many, but I just shortlisted two, dynamic bind, difftalk. I use these tools, online one. So this is also very nice docking tools. They are based on AI tools and machine learning tools. They give you, they have a like docking into the pocket, docking into the pocket, docking into the pocket. They have a several poses in the same pocket. So this is one of the AI based and machine learning based tools. There are several tools. For MD simulations, what is the definition? The definition is to simulate the system, right? To check the atomic properties, to calculate the molecular motions, right? So it gives you behavior of molecule in various conditions. What are the condition? So basically, this is not the tutorial session, but I will just go through the slide quickly, right? So what in like Atika, or she already knows the MD simulation, but for some of the students, maybe it's a new. So I'm just going to show you. So this is one of the protein you're going to target, right? Any protein. So what we have to do in in MD simulation, this is like a kind of a static molecule. So this protein is in our body, right? In our cell. It has water, right? So it has a specific space. So we draw a space around the protein, then we add water because in our system, in our body, this protein is interacting with the water molecule. Then we add the salt to neutralize the system. And then further we add whatever you want. Like in our case, in one of the study we are working now, like we are checking the role of aspirin, right? With the one of the target, one of the protein. But we are also checking certain osmolyte to avoid the side effect of drug. So whatever the study is, you can also add different osmolyte, different molecule. If you want to study the denaturation of protein, you can add a different, like urea, guanidinium chloride, and other molecule to study the denaturation pattern of the protein. So in one of the study, we added osmolyte. Okay, then further, there's equilibration, minimization, and MD production phases that this is not, we are not going to show here. So the thing is, another application is using computational tools, we can study the protein folding. So you must be knowing, you must be like thinking why we have to study the protein folding, and how it is related to the pharmacist, So see, if you see here, most of the target, of a drug are protein molecule. If you see here, probably 20% enzyme, sorry, yeah, 20% is enzyme, more than 25% is G-coupled protein, receptor. This is also a protein. So if you see, the most of the target for drugs are protein. That's why it's very important to study the protein folding. This is the protein folding funnel. It starts from the single amino acid, simple chain, to discrete folding intermediates. This is the native structure at the bottom with the least delta G. So it's very important to study the protein folding, right? From C here. So for the protein folding, once it folds, right, it acquires the very compact conformation, the least delta G conformation at this bottom. Okay, so you can also study the protein folding and unfolding using computation tools to further optimize your drug. Okay, to further predict the better docking process, right? So here, if you see why we study, because see, it also, why it is important, because it mimic the stress condition of cellular environment, right? Suppose heat shock. What is the effect of temperature on this protein? What is the effect of temperature on binding of this drug to the protein, right? You can study, effect of temperature, effect of pH, right? And it is also related to the disease, okay? Moving on. So one of the protein that is aurora kinase B, we recently explored in 2025. So it belongs to serine threonine kinase family. Okay, so it's the key component of CPC, and it also play a crucial role in mitosis. So what it has been found that dysregulation of this protein leads to abnormal mitosis. Okay, that means it has overexpressed in multiple cancer like breast cancer, pancreas, and lungs, reproductive organs, overexpressed. So what we have to do, we can consider this one as a promising therapeutic target for cancer therapy, right? So, so if we understand the folding dynamics of this protein stability under a stress condition, it may aid in drug design and target therapy. So we studied what we did. We used different concentration of urea. We used different concentration of guanidinium chloride to see how this protein unfold. And we also performed the refolding, of this protein. So here, if you see from the one of the wet lab kinase activity assay, we use 0 molar of urea. So if you see the activity is good, we added 1 molar of urea, 2 molar, 3 molar, and further we explore further. So what we can see, once you add urea, and this is the linear equation, the protein started unfolding, the kinase activity decreases, right? So here we perform MD simulation and we saw the fluctuation. So if you see RMSD, I can tell you just one line introduction about RMSD. RMSD is basically how much a structure is deviated from the native conformation. So in case of this aura kinase B, the black one, this one is your native conformation. When you add 2 molar urea concentration, the structure slightly deviated. When we add 4 molar, 6 molar, 8 molar urea concentration, the structure deviated much, right? So we can say once you add urea, the native conformation is no more a native. It deviated much from the native conformation, from the parent molecule. We also calculated the radius of gyration and solvent accessible area. So what is solvent accessible area? It will give you how much surface of the protein is accessible to solvent, indirectly or directly related to the protein denaturation. And you can report all these fluctuations of atom. This is just example of using urea. You can also use different condition, different osmolyte, different chemical molecule if you're interested, different pH, different temperature, different environmental condition, so you can explore it, Okay, so you see from the 2 molar, 0 molar, the structure is good. 2 molar started denaturation for probably 6, 8, almost teenager, right? So you can study this one. So if you see also from the computational tools, you can calculate the detailed atomic properties, detailed secondary structures also. So see here in this case, at 0 molar, 155 amino acid of this protein participated in average structure formation. When you add 2 molar, 122 amino acid. When you add 4 molar, 113 amino acid. So basically, the percentage of, secondary structure reduced. So you can see detail. I'm not going through one by one, but you can see the coil is increasing, beta sheets is decreasing, So sometimes you can do further analysis, like see in case of 2 and 4, the beta sheets are almost same. So you can say that, 2 molar and 4 molar has no much effect on, the beta sheet. So it's you can calculate the detail about the alpha properties, 310 helix, 5 helix, all these properties. And also free energy landscape, you can calculate, between the RG and RMSD. So if you see 0M, this one, so you can say it has a single deep blue color, right? So it's a kind of single well-defined energy basin, right? So a stable native conformation is here. So once you increase the 2 molar, so it has multiple lower base, 1, 2, the multiple lower base basin. It means the localized structure rearrangement is the kind of partial unfolding. If you see 4M, the two distinct, the base 4M, it means increased heterogeneity and structure expansion. So also you can check for 6 molar and 8 molar. So how, there's a free energy landscape differences. So in conclusion, we have expressed this protein and purified it. We have studied the UV and CD fluorescence, and we also found using MD simulations, that how the protein fold, right? How the urea affecting the 3D structure of this protein. And also, in our study, one of the study, we studied the binding interaction of some of the inhibitor against the COVID-19 M protease. That is the FDA approved drug, right? So we selected some of the protease inhibitor of COVID-19, and we perform MD simulations and all the detailed calculation about the binding pattern, how these drugs are binding, where it is binding, whether it is stable or not. And then from this, folding patterns, we selected the least delta G structure here, from here, the deep blue color. This coordinates, this coordinates, these coordinates. Okay, here we selected and we downloaded this structure and we saw where it is actually binding. So basically, what is this? This is an extension of docking work. You have a docking, simple docking, okay, drug fit into the pocket. But this one will give you the accurate pose, the least delta G, the most stable pose. So suppose sometimes when you have 3D structure uploaded on PDB, crystal structure, so you can also perform this, you can also calculate this lowest energy coordinates to show them that this is the most accurate binding pose of the drug. Okay, we also validated this one also. Now, this is one of the very interesting slide. So I made it very easy. So you heard about the COVID-19, right? So this is, the top protein is called a spike protein. Okay, spike protein. It's a very complex protein. See, a spike protein. It has a receptor binding domain, signal peptide, N-terminal domain. Okay, there's the different domains, S2 domain, S1 domain. Okay, this is a very complex protein, a spike protein. So you remember when COVID first appeared in winter, right? Winter 2019, probably in December. So many people thought that the virus will fade away, disappear when summer will arrive, Many people have this assumption. But when the second wave hit, it was in summer. Okay. So, so it means that this virus, SARS-CoV-2, can survive up to high temperature. Okay. So this is a very interesting one. So do you know what is the hottest weather in this world? So I was just Googling it. The hottest weather was reported 56.7 degree, probably in California. Okay, on 10th of July, 1913. The hottest weather, like less than 60. Don't get confused with another article like surface temperature in Xinjiang is 80 degrees centigrade. This is something different. Surface temperature, maybe the surface have different rocks or different metals, Surface temperature is different. I'm talking about the environmental temperature has reported. The maximum is 56 in 1913, maximum one. So what we did, we used this spike protein and we ran simulation for 0 degree, like cold condition, 20 degree, 40 degree, and 60 degree. Okay, so we want to see whether this virus will disappear in hot weather. What is the effect of this temperature on the spike protein? So we checked all this. We performed MD simulation, and we saw, if you see from here, the most functional part of the proteins are alpha helix and beta sheet. There are two. If you see alpha helix here, 21 to 21, alpha helix has no effect. This temperature has no effect on alpha helix. No effect at all. If you see beta sheets, okay, 30 to 20 is not very, not difference, So this protein is not getting unfolded. If you see another protein and do MD simulation, okay, you will see a lot of beta sheets just become loop, coil. But in this case, you see alpha sheets and beta sheets are not much, affected here. So we calculated all these RMST properties and we saw, different fluctuations and all. So we found that, the black is okay and the red, the most stable conformation. If you see green and blue, there are more. But if you see 20 degree is most stable. We can say you know, we can also if you run this computation tools, and you can see the which temperature is the most suitable and most at this temperature which the protein is stable at which temperature. From our study, the 21 we reported. So whenever you have to do calculations, you can use this temperature for all the calculations. Okay, then we'll also see the the hydrophobic part and hydrophilic part that exposed to the solvent. Okay, so these are the detailed properties, so I'm not going to detail into it. And also the spike protein, one of the study I took from one of the paper in this slide. So they only check the receptor binding domain, and they see at 40° and 50°. So they calculated that a spike protein is more stable at 10 and 50. They calculated using different tools, 10° and 50°. So they also check, this spike protein receptor binding domain has two conformation, open and close. So from MD, you can also calculate the open and close conformation of receptor binding domain. Okay, so if you see the detailed gypsy energy pattern, how it is affecting, 0 to 20 have almost similar, and 40 to 60 is 0 to 20 is similar, 40 to 60 are similar, right? See, so there's no much difference, 0 to 20 is okay, is a different, 20 to 40 is a different. So you can see the effect, on gypsy energy landscape. One of the interesting concept we recently came across. So one of our colleague, he used some chlorinated compound, during the COVID. He put some compound that is called chlorhexidine and benzyl alkonium chloride. He used two compound, two chemical molecule on the mask, and he distributed to the COVID-19 patient. So he saw using this chlorinated compound mask reduce the COVID-19, reduce the further infection. If they compare with the other individual who are using the simple mask. So, we collaborated on one of the projects. We use the receptor binding domain of a spike protein of the COVID-19, and we want to explore whether this chlorinated compound directly disrupt the membrane, or maybe it's interacting with the spike protein, So we did all the calculations about this one. Okay, also. In recent study, we also targeted, we from our chemistry department, we synthesized one of the molecule, phosphenic molecule. There's 4 different enantiomers, SSRS, SRSS, SSSS, and SRRS, right? So 4 same molecule, but different enantiomers. So this is the inhibitor of HIV proteases. So we explored the whole pattern of, hydrogen bonding, why these 4 enantiomers having the different binding poses, right? We also saw the dynamics cross correlation map, and we saw the important residues, important residues, and after binding these drugs, how it disappear, where it is binding, okay? What is the possible poses? We did all these dynamics correlation map for this one. So using computational map, you can also explore the important key residues, that we should inhibit, we should target. Okay. Another application is one of the protein is called bacteriophytochrome photoreceptor. We call it BPHP2, Ps, right? So this is red and far red light sensing protein, right? So it has 3 domain, PAS, GAF, and Phi domain. So we did a lot of substitution, 12 substitution at GAF domain, 3 substitution as this domain, right? We did mutation, and we saw the effect of binding of biliverdin to this pocket. Okay, so this is a kind of protein engineering. Okay, so you can modify the residue to check the binding pattern. Another one is, I'm just showing it quickly to show you the application of computational tools, right? So here, in another work, this is called, Sly disease. Okay, Sly disease. So he is the William Sly, Professor William Sly. He described the first patient with the Sly syndrome. So don't get confused with the, Down syndrome. This is very different. Okay. So defective gene responsible for this Sly syndrome is located on chromosome 7. So this protein beta glucuronidase. This is the protein beta glucuronidase, right? It has 49 experimental validated mutation. Okay, 49. Okay, so you can, using computation tools, you can explore one by one which is the lethal mutation, which is lethal mutations. So you can see there are the 49 reported mutations. So you can explore one by one using the computation tools, using the bioinformatics, okay, to ease your burden. So suppose out of the 49, you know, mutations, you can check using computation tool which one is more lethal, and not choosing 49, you can select only few, like 4 or 5, to work in your lab, right? To further validate it. So with together with William Sly, we published one of the, article like large scale analysis of mutation landscape in beta glucuronidase, a major player of mucopolysaccharidosis. So this is the Sly disease called common one. Okay, so he was the one who first described the patient with the Sly syndrome. So basically, moving on, so you can do further mutation engineering, enzyme engineering and mutation analysis also using these computation tools. In another study, we found that in one of the serum albumin protein, we found that this arginine 155 is very important in case of serum albumin protein, okay, of human serum albumin. But there's a reported mutation, we did a lot of mutations on different residues, but the effect was very less. But when we mutate this one, it is near the active pocket. We change arginine 155 to alanine or glutamine. We change it, right? So we have studied the Ramachandran plot pattern also. Okay, how it is affected. So Ramachandran plot, we already know it has a specific region that are permitted region. So we want to check whether after mutating these residue, are they in still Ramachandran plot, permitted region or not. So from the mutation analysis, you can do a lot of things. You can improve the property of the protein, that is called protein engineering or enzyme engineering by mutation. You can validate those, mutants. Okay, a lot of mutations, 49 mutations in this disease. You can validate, you can also give hypothesis which one is more lethal, which one is less lethal. From the mutation analysis, you can also check the dynamics pattern or Ramachandran plot analysis, right? Whether they are still in permitted region or non-permitted region. Okay, these are the application I'm telling you. Another one, in case of lipase, this is one of the lipase protein, right? The green one is catalytic pocket, and this one is called lead domain. What is lead domain? Lead domain cover the active pocket of lipase, catalytic pocket of lipase. So basically, when lead is on the top of active pocket or catalytic pocket, that means it is closed conformation. That enzyme is not active. When lid is not on the top of catalytic pocket, that means the active pocket is exposed or open. So it is active lipase. Okay. So we find a lot of different lid, lid molecule. Some lid is just alpha helix. Some lead is like just a loop. Okay. Some leads are very short alpha helix. We try to delete those leads. We did a lot of mutations. We try to replace the lead with loop, right, without affecting the structure. And we also studied in MD simulation the open and closed conformation. Okay, we calculated everything, the distance and all. So this is very interesting. Like suppose this is the lead, right? You want to activate this protein molecule for long time. Okay, you don't want it to be inactivated. So you can replace this lead by just a loop without affecting the structure, or maybe having the larger lid, okay, so that the pocket is partially open. So you can do enzyme engineering using the computation tools. So here, so these are all the, so I did not add about the AI. Nowadays we have several AI tools also, like dynamic bind, diffdoc to calculate the binding pose. There are several AI poses also. So I did not include here, but I just included in the future one that future sustainable drug discovery lies in the integration of advanced computation techniques. It reduces the cost, waste, and ease our experimental burden. Okay, so generative AI is possible, like enable de novo molecule design and predictive therapeutic development. And also the the quantum computing, it promises high accurate molecular simulation and interaction predictions. And some tools like digital twins support the personalized medicine and reduce trial and error approaches. So another one is autonomous drug discovery. It combining with AI robotics and self-optimizing competition pipelines, it transforming pharmaceutical research into faster, greener, and more sustainable process for next generation therapeutics. So this is just kind of like, future of sustainable drug discovery using AI, quantum computing, and the concept of digital twins or autonomous, self-optimized pipeline. Okay. So personalized medicine, these are the concepts for future sustainable drug discovery. I did not include. They already, there are a lot of work related to AI, AI prediction, but I did not include here. I just kept in future one. Okay. So what are the conclusion? The conclusion is our computational tools, the bioinformatician tools or medicinal chemistry tools, you can say, the computational chemistry discovery is rapid transforming pharmaceutical research into more sustainable and environmentally friendly responsible discipline by reducing the chemical waste, laboratory burden, and development cost and energy consumption. Computational approaches strongly support the principle of green pharmacy. Tools like molecular modeling, virtual screening, ligand-based virtual screening, enzyme engineering, simulations, these accelerate the therapeutic discovery. It also improve our mechanistic understanding of disease, like folding, unfolding pathway, and it also enable more rational drug design. And at the same times, it advances in artificial intelligence and computational biology that are driving sustainable pharmaceutical innovation. And it is for the future precision medicine, shaping the future of next generation healthcare and therapeutics. So basically, in the future one, we are hoping that generative AI and this quantum computing, along with personalized medicine, will ease our burden. Will leads to a more clean and green. So this is the conclusion I added, and hopefully I completed my presentation before time. I would like to acknowledge, still I'm using the South African CHPC and CHPC in our
Oh,
university.
okay.
Yeah, because we have different projects, in collaboration. So I'm still occupying that one. Yeah. Thank you so much.
Right. Yeah, thank you so much for
Mhm.
the really interesting Dr.
Mhm.
Khan for
Yeah.
your talks. Yeah.
Mm.
So we actually already got some questions here in the chat box.
Mhm.
Yeah, and I think one of this from a participant here,
Mhm.
she kind of asking about the simulation time. Is there any like a
Yeah.
standard or minimum requirement for that?
Yeah. See, I'm frankly speaking, in 2012, right, in South Africa,
Mm.
the Gromax or Amber was such a new tools, simulation in
Mm.
2000. I'm talking about the 2012. So nobody knows how to run simulations. I was joining workshop in University of KwaZulu-Natal and Cape Town to learn from, so I was going there. That time to check the RMSD 10 NS was okay to publish a paper. But frankly speaking, if you see enzyme kinetics, they are in seconds, not in nanoseconds. So simulation, if you have computation, if you have a good computation platform, just use minimum 500 NS or 1000 NS. That's all. And also, what is the minimum? This is no correct answer for this question. I will just tell you one thing. Just I will tell you one thing here. Okay. Can you see this one, right?
Yeah.
So we run for 200 NS, right? 200 NS.
200.
Yeah. Because this is denaturation study, right? So if you see this black and red, don't look other one, right? Because this is denaturation, it's a fluctuation. If you see the black and red, I can say this 200 NS is okay. Why? Because the system is equilibrated. A straight line. When you see a straight line after 20 NS or 50 NS, you can see, okay, it's a good for analysis. But if you have a good computation, platform like CHPC, high computation facility, run 1000 ns, 500 ns. But if you get a straight line, enough for analysis. Because in real time, they are not in ns. Enzymatic activity is in seconds, so you need more time. But if you see a straight line, enough for analysis. Yeah. The reviewer is never happy with your results. If you do 200 ns, he will say 500 ns. If you do 500 ns, he will ask for 1000 ns, So just defend your analysis, I can see a straight line. It's fine. It's enough for analysis. Yeah. So there is no proper answer for this question, More is better. More is better. Yeah.
Yeah. As long as we check for the RMSD, right?
Exactly.
Yeah.
It's a
I
straight
think
line. Yeah.
straight line.
Mm.
Okay. And then the second question, it's about, is there any like program, software package that can be run in a laptop? Because I think, yeah, participant thinks that it needs special requirements like workstations, a special one like CHPC in South Africa.
Yeah. See, I'm telling my personal experience. I'm not very core, programmer. Okay, I just use Google if I get error. But my personal experience is when you use GPU, not CPU. When you use GPU is much faster, much faster. GPU. Regarding the workstation, obviously laptop is not good. But what you can do, my personal experience, do all the analysis, not the calculations, up to the last step before MD on your laptop. It's faster. Okay. And then finally, there are a lot of collaborator. Every university now have CHPC nowadays. So you can collaborate. The easy way is to collaborate. No need to buy workstation. Just collaborate. If some of your PhD friends are in the audience, definitely you can collaborate with us if you have some good experimental data. Because I have 4 PhD students, probably we can assign some work to them, Good to collaborate, to learn and collaborate, because all the university nowadays have CHPC, but use GPU. Yeah.
Okay, Dr. Hans. So
Mm.
the GPU, maybe you can mention like what is like the example for the GPU itself. Maybe some of the
Yeah.
participants here are not familiar with that. The like
Yeah.
NVIDIA things.
Yeah. So for the two nodes, GPU and CPU, right?
Yeah.
So how to say? I cannot answer this question. I already told you, I'm not very core programmer or the computer background. It's still a biologist, bioinformatician, but biologist. But my experience is GPU is faster when I compare it. What is the reason why GPU is faster? You can just Google it, and I have to Google it also. Yeah, but
Sure.
I'm just sharing my experience. And regarding
Okay.
if you don't have any, CHPC, just collaborate with any one of them. I think many universities, even in Indonesia, Malaysia, they have, they have
Yeah.
workstation. Yeah. Even I bought last month, I bought one workstation, and still I did not touch it. I told my student maybe from the next month, because the next month teaching ends, so we are going to explore that workstation. So nowadays it's very easy, to just know where is the workstation, collaborate with them. That's all. Yeah.
Yeah,
Yeah.
sure. I think in Indonesia also we have the like institute, national institute that can provide that as well. I think
Yeah.
yeah,
Mm.
for those in
Yeah.
Indonesia, I think. Yeah. Doctor, and about the software package or the program, do you have any
Yeah.
recommendation for the beginner?
For the see, before maybe you also know, before people were using, I should not say this one, right? I should not just say this one, but people were using Amber, and they're very
Yeah.
happy with the Amber. We were also using Amber because in CHPC there was Amber and Gromax, but all of them switched to Gromax. Gromax is
Yeah,
free,
I
free.
think so.
They
Yeah.
have more advanced properties to calculate. Okay. And everyone using Gromax, if you find any error, just Google it, you will find the solution. If there's an atomic clash, you can find the solution on Google. There are a lot of discussion going on. So I will suggest you, even if you have Schrodinger package, use it. Okay, Schrodinger package, use it. But I'm saying Gromax, easy, free, And any errors, just Google it. You will find a lot of discussion. So frankly speaking, I got a lot of error, atomic clashes. The water molecule is not equal to what you mentioned in the text file, topology file. I Google it, how to remove it, how to remove water molecule, how to add more. I managed to do it. Best is Gromax. My experience. Yeah. Mm. Yeah.
Yeah, the
Mm.
Gromex one. I think that's the popular, I think worldwide for Dr. Khan, yeah.
Yeah.
Gromex, and also lots of publications already
A lot.
released. Yeah, lots of that using
Yeah.
Gromex as well.
Mm.
Yeah, Dr.
Okay,
Khan.
okay.
Yeah.
So for your, for your question, can you see the slide? So see,
Okay. Yeah.
yeah.
Maybe
So it's
anyone here
yeah,
can check the
yeah.
share screen.
It says, why I asked why GPUs are faster. They possess thousands of small cores for parallel processing and all these computer things. Okay, so yeah. Mm.
Yeah, okay, that's the answer
Yeah,
anyone is
exactly. Yeah. Mm.
Yeah, I
Mm.
think that's a really
Yeah, yeah.
valid resource.
If you present your simulation work, I will ask same question. So yeah. Mm.
Okay, doctor. So anyone here would
Yeah.
like to ask another question? Maybe you can raise hand or another question in chat box. I think I saw one of the raising hand here. Yeah. Any more question? So maybe I would like ask question while waiting for the other
Yeah,
questions from the participants. Yeah.
I think one of them asked about the 50 NS round, right?
Yeah. 50 run.
Yeah, I explain, if you have
Hmm.
computational resources, extend more, 500. But if you don't have like 50 NS data, okay, this is one way I will tell you. Suppose you have a good enough data. I have time, right? I have time, right?
Yeah, sure. Yeah.
Okay,
I think
okay,
we will finish on the
okay. So I
10.
will
Yeah.
I will tell you one thing. The best way. Suppose you have a good data. Okay, you have a good pharmacy data, like lab data, right? And you did MD simulations. Okay, only 15 is because you don't have computational facility, right?
Yeah.
Once you submit a paper, see, I'm the reviewer, you are the reviewer. You will first comment it. Why 15 is? This is a very common one. So the best way to avoid is because the simulation is not the main part of the paper. Best ways to avoid it, don't mention the time scale. Mention this one. Do clustering, download the lowest coordinates and show the best binding poses. Don't show them the time, this one. Don't show them this RMSD and RMSF. Okay? Just
If there's
say,
question about
yeah,
that.
yeah, no, yeah, exactly, to overcome,
Yeah. Okay.
yeah, because even if you get a straight line, the reviewer say, no, we want to see more. So best way, don't show them RMSD, RMSF. If you are satisfied with your straight line, the result, just use the lowest coordinate, the least delta G structure, and show them the binding pose if you're working on drug. That's all. This is enough. Yeah, this is
So
enough.
using scoring coordinates here, doctor.
Exactly, yeah.
Okay. Yeah. And how about the RMSF, doctor? You mentioned earlier
Yeah,
how it
so
helps. Yeah.
I don't think RMSs they show the time scale. Let me see. Where's RMS? Yeah. So this one, RMSD they will show you I think the time scale, right? So bring to the front. Okay, so this one if you see for RMSF you can show the fluctuation. It is RMSF fluctuation versus residue. So it's okay. It will not show you the time. It will show you the residue that are more fluctuating. If somewhere is the active pocket, you can show them, this residue have least fluctuation after binding of ligand or more fluctuation after binding of ligand. I don't think this will give you the time scale. So it's fine. This one you can include it if you are satisfied with your MD. You can include this one. You can include this the lowest coordinates.
Yeah.
Yeah, to avoid to show, showing them the 50NS and all. Yeah.
Okay. Now we already got the
Mm.
other questions, doctor. So from Ibu Masito, yeah, Mrs. Masito. So as far as I know, running AI and machine learning use considerably amount of energy. And what can we do to make computational drug design greener energy-wise? I think that's related to how we utilize the computer itself.
What is the question? Where is the question?
That's the what can we do to make computational drug design greener, like a green pharmacy? So it's like how the carbon footprint reduced by using the computer. Does it have any
At least
impact
if
on that?
see if you
Yeah,
if you
yeah.
compare with the chemical waste, you know, if you compare with the chemical waste, if you compare with the time, right?
Yeah, I think
So
that's much, much better
it is,
using.
yeah, it's much
Mm,
better, yeah, much better, yeah. So this is the
yeah.
only, yeah. Obviously, electricity and all is not a green at all, but it's still, yeah.
Yeah, sure,
So
doctor. So
if you compare with the time and the chemical waste, so it's still better.
yeah,
It's not a
using
very
chemical.
yeah.
Yeah,
So frankly speaking, before the computation tools was not so accurate, but nowadays, like if you at least report 5 molecule and validate it, obviously you will get two corrected. The better one. Yeah.
yeah.
Mm.
Okay. Actually, the one who asked the question
Mm.
next to me. So I think, yeah, he already answered the question. Yeah.
Mm.
Okay. And then another question is about the last one in the chat box. Current
Mm.
AI. Oh, there's another one pop up.
Current
So
AI discovery.
yeah, do you think future pharmaceutical innovation risks becoming technologically advanced but biologically oversimplified? I think that's a good question. What biologically
So yeah,
oversimplified?
AI-driven drug discovery platform can generate and optimize molecule very rapidly, but biological systems are highly dynamic, complex. Do you think future pharmaceutical innovation? No, no, no. See. Never rely on computational outcome. I'm not saying that these results are perfect. I'm not saying, but at least from the thousands of molecules, you are reporting two good molecule, five good molecule. Out of five good molecule, you may get one good molecule when you do experiment. Okay, so I'm not saying this is 100% correct. Regarding the current AI process is quick. For example, one protein has several pathways, right? Pathways. So one protein is interacting with another protein, right?
Mhm.
If you see the receptor, GPCR receptor, if you see the diagram, so it has several mechanism. It is going through, you know, several ways interacting with other molecules, and then finally going to have a PPI with another protein molecule. They are having just several obstruction, in this process, in biological process. But here in computation one, what we are going to see PPI, we just have two protein.
Oh
We
yeah,
don't have
protein
any,
protein.
yeah,
Yeah.
you don't
Mm.
have any other things, in the middle of this two protein. So that's why it's very quick, And I'm also saying never rely on these tools, It's just a kind of prediction, hypothesis, how it can bind, like allosteric site, If ligand bind to allosteric site, is there any change in the pocket, at the real pocket? So once you add many drug, you may find the different size of the pocket. So you can assume that it's having some change or no changes. You can give hypothesis. It's never rely on alone on computation tools. I never said that this is the most accurate result. You need experimental, validation.
Mm.
Yeah.
Yeah, interesting. So the other question, Doctor, from your perspective, what
Mhm.
emerging areas in computational chemistry are expected to have the greatest impact on the future drug discovery?
So what emerging areas, right?
I think the
Computational
most
chemistry.
one.
What should I answer of this question?
I
Emerging
think
areas.
yeah,
Yeah.
the most emerging one.
So what I'm saying, emerging one, most emerging one. So basically, you know, I work on protein,
Mm.
So I know
Yeah.
about the protein molecule. Yeah. So emerging area, I don't know. I don't know how to suggest, better emerging area. What is your interest, It's the best emerging area,
Yeah.
Yeah.
I
Yeah.
think maybe Rahmatun Nafiah, the participant who asked the question. You
Uh-huh.
can hello. Maybe I will grade her in
Yeah.
bahasa dokter. Ya.
Yeah.
Halo Bu Rahmatun. Mungkin bisa lebih perjelas ini untuk di bagian apa dari computational chemistry yang ditanyakan. Halo Bu Rahmatun. Bisa langsung unmute, mungkin bisa sedikit menjelaskan. Dalam bahasa Indonesia tidak apa-apa, nanti saya akan bantu.
Okay, I can see the last question also.
Yeah.
Yeah.
I
Yeah.
think, yeah, it's not that thing.
Yeah. This question is very interesting, you know, the last question by in the. So
Yeah, Indah.
the two question, to what extent can molecular docking and molecular dynamics studies can consider reliable without wet lab? See,
Yeah.
if you want for paper point of view, you know, for paper point of view, some paper you can publish up to 8 impact factor without, you know, without having the wet lab. But the best way, if you have some data from the wet lab experiment, you know, you will just you know, add more eye-catching figures to the paper. You know, you can just support those wet lab outcomes by graphics, you know, the colorful figures, you know. So basically they are publishing recently. My master student published 5.8 impact factor just for simulations one, you know, 500 simulations. So now see simulations, it all depends on the quality of simulation. For example, here, just I'm telling you, can you see this one RMSD, right? One black. Nowadays, they are not accepting this just one RMSD. They are accepting replica. So one MD, you can use same condition 3 MD replica, and you know, and give the results like average results, like what we do in experiment. So if you report like 500 NS up to 500 NS with each simulation in 3 replicate, okay, you can go up to, you know, high impact one, you know. And also, how can an in silico study remain candid and competitive for publication in high impact journal without I never said use without experimental data. If you don't have, you know, experimental data, try to you should not rely much, but at least you have some hypothesis. You can just propose something, you know. And whenever you write, you never say that we found this drug to be more active. No, we predicted this drug to be active, you know, whenever you use computational tools here. So nobody can, you know, question your results, you know. You wrote predicted. Prediction can be wrong and right. Okay. Yeah. In our institution, we currently learning computational drug discovery methods such as molecular docking, Lipinski rule, admit prediction, but we still have limited experience.
I
Could you
think that's
recommend
already
some
answered it.
Yeah, so Gromax, definitely Gromax. And
Oh,
also,
besides,
yeah,
okay.
and also when we, frankly speaking, nowadays in our CHPC also, Schrodinger is very nice. You know, before I was using Discovery Studio, but Schrodinger package is having
Schrodinger,
everything.
yeah.
It's a bit expensive, but
Yeah, but
everything. Yeah, but it
it
has
needs
every.
license, yeah.
Yeah, it has everything. Yeah.
Okay, Schrodinger. Yeah, and
Mhm.
also, what is your opinion on the future perspective of
Ha-
hantavirus research?
Hantavirus research in silico drug discovery. I don't know about hantavirus, you know. It's
Hantavirus.
yeah, it's up to you, you know. If you are interested in working on hantavirus, I don't know what is hantavirus. Yeah, you work it.
Yeah, I think
Hantavirus is ortho hantavirus.
let me check
Singapore.
first.
Yeah.
Yeah, it's Southeast Asian
Food
virus.
form,
Yeah,
yeah, in the rodents, right? In the
rodents.
rodents.
Yeah, rodents.
Yeah. Maybe if you're interested, just explore, you know. Do you think
Mm.
hantavirus could become important target? I have no idea about hantavirus. I just Google it, and for the first time, I, you know, yeah, I'm so sorry.
Yeah,
Yeah,
so I just heard that.
yeah,
Yeah,
but yes, but yeah, yes, you know, you are
RNA.
asking, do you think we can explore differently? Explore it, definitely explore
Yeah.
it. Yeah, natural
Yeah.
product research. Yeah, definitely explore it. You know.
Yeah, it's still
Yeah.
possible to do the same things
Yeah.
like in SARS-CoV-2, SARS-CoV
Yeah, exactly.
9
Yeah.
to this hantavirus, right?
Yeah.
Yeah.
So
Mm.
select a good target for this hantavirus. You get the database of drug, FDA viral drug, FDA approved drug, and you just explore, do high throughput screening, select it, do simulation, report it, predict it, explore. Why
Mhm.
not? Just explore.
Yeah, I
Yeah.
think that's really the take-home message for the
And
questioner here.
And again, can
Yeah.
nanomaterials, I would like to ask, computational chemistry and AI are currently being used to design nanomaterials. Okay. I think you have experience of nanomaterial, right? Or bio? I'm not sure.
Yeah, both of us.
Both of
Yes.
us, yeah, exactly.
But
So
nanomaterials, the inorganic materials, bio has
bio,
that.
yeah. So
Yeah.
bio. So next presentation is for bio. He can explain
Mm.
more. Yeah.
All right. Okay, that's it.
Besides his favorite topic, nanomaterial.
Yeah,
Mm,
sure. I
mm.
think, okay, any other question maybe from the audience?
If you want to ask about the mutation, how to do analysis of mutation using MD, I can tell you everything, how to denature, folding, refolding, all this kind of analysis, I can explain you. Yeah, but for
Okay,
nanomaterial, hantavirus, I have no idea.
okay.
Mhm.
So all about the proteins, yeah, I think that's
Yeah,
really
exactly. Yeah.
really fun
The,
for
it's still
studies.
there, it's still there a lot to do in protein, a lot to do.
Yeah.
Mm.
Also, maybe the capsid or the capsule of the virus also has the
Yeah,
protein, then
exactly, exactly.
that
Yeah.
makes sense. Yeah.
Hmm.
Okay, doctor. How about the other question? Is there anyone going to raise some questions more? Okay. Yeah, I think that's the question so far, doctor.
Yeah. Okay,
Yeah.
thank you.
Okay, doctor, maybe before we really close the session, do you
Mm.
have any take-home message for our younger generations, like going to really do the computational, or maybe going to be a computational chemist in the future?
Yeah, the thing is, these tools, computational tools, the thing is, once you enter in this field, computation biology or computational chemistry, so you have to get updated, all the time. Like GROMACS before in 2012, when we learn it, RMSD, RMSF enough to publish a paper, But you have to explore. If you see this, the analysis tool, GMX,
Yeah, Jim.
GROMACS, yeah, there are, thousands of analysis. See, thousands of analysis.
Yeah, that's
You
the
have to, yeah,
comments.
thousands of, all the
Mm.
description.
Mm.
So the thing is, you have to get updated if you're going in this field. See here. Can you see the screen? Like before, it was just RMSD and RSF. Now, see, you can calculate the angle, angle.
Mm. Yeah.
You can
Angles.
calculate the eigenvectors, bar, bundle, chi, cluster, conforms to see the, you know, you can
Lots of
also
functions there.
covariate, yeah, density map, Before it was not that much complex. So once you enter in
Mhm.
this field, there are a lot to do. I will not say that, this is outdated field, a lot to do, but you should be updated, updated what's going around.
Mm.
For example, here, I will tell you one thing. Uh, okay, I think it is in the PPT. I still have time, right?
Yeah, still true.
Okay, so just example, Mm, yeah, yeah, this one, probably this one, yeah. Okay, this one. For example, you want to add 5 molar of the compound, right? Suppose you want to add something 5 molar. Okay. Now, what we do in computer, most of the people did before. They added a protein in the center, so they calculated box size. Okay.
Yeah.
They added the water molecule. Okay, they added the water molecule, And for 5 molar, they calculated based on the box size. This is wrong. When you have a protein, when you have a certain box, when you add water and when you add some osmolyte or any molecule, it also kick out certain water molecule. So you calculated 5 molar, but it becomes 5.5 molar, in fact. So these are very important things that you should get updated, you know. So every step for competition chemistry, you have to calculate how much water is kicked out. Okay, so one molecule kicked out 3 molecule of water, you have to adjust your calculation accordingly. So this is the most important thing, you know. If you're choosing this field, you should get updated. What's going around, you know, the new tools, you know, new calculation, yeah, everything, yeah.
So basically, it can mimic kind of mimicking what happened in
Yeah,
the
exactly.
the wet lab, right?
Yeah,
So it's
exactly.
when they all the
Actual system.
experiments.
Yeah.
Yeah, the actual
Mm-hmm.
one. If we consider
Mm-hmm.
the concentrations.
Yeah.
Okay. Oh, another question just coming up. So
No, it's
maybe this
okay.
is the okay. Yeah, I think this is the last question, doctor. Is that okay?
I'm
Yeah.
new to molecular docking based on visualization. How do we know if the ligand position from the docking is correct? Exactly. This is very important. Very important. You asked, I forget to mention, you know, nowadays there's a, you know, this is the most common questions. Who is expert in this field? Docking, you know,
Yeah.
molecular docking. Everyone is doing the molecular docking. Everyone is doing. So that is not accurate, you know. So what we have to do, for example, here, mm, we have time.
Yeah. Maybe around 3 minutes.
Oh, 3 minutes.
Yeah.
Okay. So okay, it's not. Uh, oh, very nice, you know. Yeah. So 2026. Just a quick. I will show you. Uh, It's not moving. Okay, just a quick one to answer your
Sure.
question. Uh, Lecture 9, drug development part 1. Okay, here. So everyone is doing docking, right? So this is phospholipase A2. The PDB structure is 1OXR. It is already, there is already aspirin molecule inside. Okay, aspirin molecule. Suppose you found a new drug, predicted a new drug, you want to do docking. Okay, after, okay, and you got the highest score, delta G, blah, blah, minus 10 kilocal, something, you know. After docking, the best validation is just superimpose these two, you know, this one and your dock pose, superimpose. And if you see the orientation of ligand is same or not, nobody is asking further questions. If you dock it and you write a sentence, we further superimpose with the co-crystallized, you know, ligand already present in the crystallized structure. And we found that the orientation of my docking pose and co-crystallized pose is same. No more questions, you know. This is the best way to validate the docking one. It's still computation one. It's not 100% accurate, but it's still to validate the docking pose, better just superimpose with the already known co-crystallized ligand in the protein molecule. Just superimpose it.
Yeah.
Yeah,
Superimposition.
this is a pretty easy one, you know, pretty easy one. We don't have time, but there are other ways. But this is the nobody's going to ask question if you write these sentences. We superimpose it, and we found that the my dog pose and the co-crystallized pose is in the same orientation. So you got maximum delta G, you got the same pocket and the same orientation. 3 validation. That's all. Yeah.
Yeah.
Mm.
So I think we all agree with that.
Yeah.
People worldwide, yeah. Okay, maybe hopefully that's all the answers from the Dr. Han. Anyone? So if you have any other questions that want to follow up with you, doctor, would that be fine to contact you
Yeah, sure.
through email?
So,
Yeah.
yeah, so you can contact via email.
Yeah.
I did not mention, right? Email?
I think it's already present in the
Yeah,
website that it's
exactly,
open.
exactly.
Okay,
That
yeah.
should be, yeah, that should be okay.
Yeah.
Mm,
Okay.
mm.
Okay. I think it's been a sincere pleasure for me, doctor, to guide you through this session today. So thank you again for your sharing
Okay, thank
your
you for
expertise. Yeah.
your
Okay.
invitation. Yeah, exactly.
Yeah,
Thank you,
yeah, for sure. Yeah. It's been wonderful to catch up and learn again from you today.
Please,
Yeah,
please, yeah.
especially for myself. Yeah.
Mhm.
Okay. To our participants, thank you for your engagement and your thoughtful presence in this virtual space. We hope the insights shared by Dr. Han will spark new understanding, yeah, in your own research journeys. So enjoy the rest of the conference. I will return the session back to the MC, the Salsa and Grace. Thank you. Assalamualaikum warahmatullahi
Okay,
wabarakatuh. Thank you so much,
okay.
Dr. Han. Yeah.
Okay, thank you. Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Khan.
Okay, thank you. Bye-bye.
Thank you, Mrs. Atika.
Now, we would like to present a certificate of appreciation to our first speaker, Assistant Professor Faiz Iqbal Khan, PhD, for the insightful presentation and valuable contribution to this international conference. We kindly invite Dr. Apotheker Sri Haryanti, Magister Science, to join us on screen and deliver the certificate virtually.
Thank you. On behalf of the head of institution of STIFAR Yayasan Farmasi Semarang, we would like to express our sincere appreciation to our distinguished keynote speaker, Assistant Professor Faiz Iqbal Khan, PhD, for the insightful and inspiring presentation delivered today. As a token of our gratitude, we are pleased to present this certificate of appreciation virtually. Thank you once again for your support and participation in second SINACON 2026.
Yeah, thank you so much.
All right, once again, thank you for Assistant Professor Faiz Iqbal Khan, PhD, for the insightful presentation. Ladies and gentlemen, we now proceed to our second speaker, Professor Dr. Ganti Hua from the Faculty of Pharmacy, University of Cyberjaya, Malaysia.
Professor Gan will present on the topic of green pharmacy approaches in natural product-based drug design for sustainable therapeutics.
We will invite Dr. Apotheker Endangwi Wulantari, Magister Science, as the moderator for this session. Mrs. Dewi is the head of the Magister Program of Pharmacy in Sekolah Tinggi Ilmu Farmasi Yayasan Farmasi Semarang.
For Mrs. Dewi, the time is yours.
Check. My sound's clear. Okay, thank you to MC. Check. Okay, good morning, distinguished guests and all participants of the second SINACON 2026. In this second session, the presentation will be delivered by Professor Gansu Hua. Hello, how are you? Good morning.
Good morning.
Apa kabar?
Baik.
Okay. Nice to see you again, even if it's only in virtually in the Zoom meeting.
Okay.
Okay, this is a great pleasure for me to be your moderator in these sessions. Before we begin, let me share, read your brief profile of these second sessions. Our amazing speaker in these sessions, Professor Gan Sil Hua. Sorry for spell it maybe the wrong spell. I apologize. Professor Gan Sehua complete her pharmaceutical studies at the Manchester University, England, and she then obtained her clinical master of pharmacy and PhD degree in pharmacology from University Science Malaysia. Prof. Gan's work experience began in 2003 as an assistant lecturer at USM, correct, Professor?
Yeah.
And
Shows how old I
Professor,
am.
oh no,
Oh
you look
no.
younger. Okay, Professor PK7 at USM in 2014 until 2018, and Professor at Monash University in 2018 until 2024. And now she is the Dean of the Faculty of Pharmacy in Sabarjaya University. It's almost a year since we met at your campus, Prof.
Yeah. You acted very fast.
Yeah, and we are getting older. Okay. Uh, Focus on pharmacology, pharmacogenetics, toxicology, and then pharmacokinetics, natural products. She has hundreds of publications in high-impact journals. There are more than 300 articles, 30 proceedings, 30 books or chapter books, and many experiences as an invited speakers, and also granted and awards. Yes, so many. There's a long list of your CV that I can't read all of it. Therefore, it is no surprise that she was included in a Stanford University list of the world top 2% scientists from 2021 until 2025 consecutively. It's a very consistent professor for 5 years.
I understand.
Right, very impressive.
Thank you.
And in these sessions, Professor Gan will share her knowledge with us for about 50 minutes, Professor.
Okay.
Okay, and then we will continue into question and answer discussions for about 10 minutes. Okay, let's warmly welcome to Professor Gan. The screen is yours, Professor.
Thank you, Devi. Namo Buddhaya,
you.
everyone. Thank you for the invitation. I will share my slide. Um, let me see. Can you see the slide?
Oh, yes, Prof.
Yes, sir.
Okay, so I will start then. So, good morning, everyone. Thank you for inviting me here today. So, I'll be talking about green pharmacy approach in natural product-based drug design for sustainable therapeutics. However, because my research is on honey and propolis, I will use them as an example. I hope my voice is clear.
Clear, good.
Okay, so this is the content. First, I will give you the meaning of green pharmacy, and then I will talk about honey, the different types of honey, and how honey is being characterized, and how do we ensure that the honey is pure, madu. If the Indon say madu, madu tiga, is it? And then a little bit on medicinal properties of honey. I did a lot on this, but I'll just show you one. And then I will touch on propolis and how we produce a green product from propolis together with the industry and the challenges of green pharmacy. Yeah, so basically that's what I will cover. So green pharmacy covers 12 different areas. Some of it is like prevent wastage when you conduct scientific experiments in the lab. Do not create unnecessary waste. And then use less hazardous chemicals, use safer solvents, energy, sorry, wait, let me move this. Energy efficiency, and then use renewable raw materials, meaning that the beginning raw material must be sustainable. And then there are many factors you can see here. So this basically is what meant by green pharmacy. So in other words, it is integrating ecological sustainability or continuation into the entire life cycle of natural product-based drug design, starting from the source, the sumber, to the disposal, how we remove them, by applying green chemistry principles, the 12 principles I showed just now, and by leveraging on modern technology that we have now to minimize environmental impact and to promote long-term resource availability. So we don't want to use up all of the things on Earth and leave nothing to our generation. So basically that is the aim. So first I will give the background about my honey research, and then we will see about how green pharmacy can be applied to both honey and also propolis research, which I will share later on. So bear with me if this topic is not within your area, just get it as a general knowledge. So there are more than 300 types of honey in Malaysia, Indonesia, everywhere in the world. There are more than 300 types of honey. And normally we will compare it with Manuka honey as the gold standard, not because Manuka honey is superior, but because it is widely researched on a lot of data on Manuka honey. So people use that for comparison. So do you know when you consume honey, you actually consume the flower nectar and the bee saliva? So what happened is the bee will collect the nectar and then will swallow it and then churn it in the stomach and then spit it out and then swallow again and churn in the stomach, spit it out for at least 4 to 5 times. So when you consume honey, you actually consume the bee saliva, which is good in this case because it has a lot of amino acid and vitamins and other nutrients. So as you can see, the different types of honey is dependent on the type of flower that the bee visit to. And at the bottom here, on the bottom left, the brown color one, we have a special type of bee, which is called stingless bee, which do not sting because they do not have sting, but they do bite, and it's quite painful also, just like an ant bite. So we have 2 different types of bees. The big bee, which is about 10 times the size of the small stingless bee. So this is significant because later I will talk about the sustainability of this stingless bee, because we can rear stingless bee along the corridor at our house and so on. And this fits one of the green pharmacy aim of ensuring sustainable raw product. So when we deal with stingless bee, we can have limitless source of honey and also other products. So do you know that honey is about 80% of honey found in Malaysia is fake? It's not real, meaning that it's sugar water. It's very easy to fake honey because it's mostly widely used sweetener. And about 75% of honey is fake in the Philippines. I don't have the data for Indonesia, but I suspect it's very similar. And honey is the world's 3rd most fake food. is found everywhere. And so I talk about Malaysia since I'm in Malaysia. So Malaysia, just like Indonesia, being a tropical country rich in its flora and fauna, has many different types of honey. And honey has been used for ages as wound healing, antimicrobial, and also anti-aging, for its anti-aging properties. And I'm sure in the Quran also, in the Holy Quran also, it does mention about the goodness of honey and studying honey. So our first objective in our beginning of the research is to whether to know if the honey consumed in Malaysia is of high quality and is pure, basically. So we look at the four things, and we randomly purchase honey from the supermarket shelf, and we compare it with Manuka honey because it's mostly researched on. And we look at the physical chemical properties, the pH, moisture content, the color. The darker colored honey tend to be better because it has a lot of pigments like flavonoid and phenolic, which contribute to the antioxidant properties or the anti-cancer properties. So if you want to choose honey, go for the darker colored honey. And then also it has vitamin C or ascorbic acid. It has proline. Proline is a type of amino acid produced or found only in the bee saliva, and therefore it must be present to show that the honey is pure. And if there is no proline, very highly likely that it is adulterated honey. And we also did the antioxidant analysis, the basic ones. And we use the International Honey Commission guide to say that our honey is, whether to determine the honey is pure or not. So the pH must be acidic, moisture content less than 20%, total sugar more than 60, sucrose less than 5%, 5-hydroxymethylfurfura, I will talk, or 5-HMF, I will talk more about this because it's very interesting compound. Color intensity, I have already touched just now. Protein and proline. Proline must be present because it's from the bee saliva. So this criteria must be fulfilled to ensure that the honey is pure. So we found that the honey samples from Malaysia were acidic, indicating that they are good, they are pure. And then acacia honey was most acidic. And the more acidic it is, the longer is the shelf life because it tends to be stable against microbial spoilage. And then the moisture content was within the less than 20% recommended by International Honey Commission. And Manuka honey, if you look at Manuka honey, is very viscous, right? Very pekat, the Indonesians say. So it has the lowest moisture content of about 12%. And then sourwood honey had very low sugar content. And if perhaps a patient is diabetic and wants to take honey, perhaps sourwood honey is something that he or she should go to. Because we also believe that traditionally for diabetics, you should go for sour and also bitter food, right? So these are one of the tips that I am sharing with you all. Honey from India tend to be very sweet. And the sucrose content for Malaysian honey is also within the recommendation, which is good news. And this is the compound that I want to talk about just now, the 5-HMF. It is found in honey. It is released during honey processing when the bee farmer, they collect the honey and they try to sterilize it, they heat it up, so it is released. Or due to poor storage, for example, you put your honey bottle near the window and then it gets the sunlight, or in the car, that's worse, or in old honey. So 5-HMF is released, the longer you store the honey, the more it is being released. And it is of concern because this 5-HMS is found to be carcinogenic. It causes cancer when it is alone. But we didn't analyze the fact that if it is inside honey, we didn't analyze, but we only analyzed 5-HMF alone. The reason I say that is being natural product, the other components present in honey may inhibit the activity of 5-HMF. So maybe that's the reason why people are still consuming honey even though it's full. But our caution is that we found that honey, when it's stored beyond 1 year, the 5-HMF content is super high or above the recommendation of ICH guideline. And we didn't analyze whether it can cause cancer or not. But because this substance can cause cancer, we recommend that honey should be consumed within 1 year of its processing, just to be on the safe side. I hope that's clear. So I'm not saying that if you take honey more than 1 year, you can get cancer, but because we didn't look at the other substance that can inhibit it, but we found this 5-HMF to be very highly present if kept for a long time. The honey is kept for a long time. So this is our article. And then I already mentioned about honey color. And again, sourwood honey tend to have darker color, also tualang honey. So for us, we find that these two types of honey are good. This is the total phenolic and flavonoid content, if you would like to see it. And then we also look at what are the specific phenolic acid present by using HPLC. And we found the constituents present. And we were the first to report on the 6 phenolic acid present in Malaysian honey and 5 flavonoid in Malaysia honey. Yeah, so if you want to be, so this paper won the top most cited paper in Journal of Food Science. So if you want to be the highly cited person, then you can go for the first. Always find the first. I'm the first to find this. I'm the first to find that. Yeah. So that's my tip that I'm sharing with the young researchers that we have here. They still have a long window to go to. And then we also look at the vitamin C content. Sour wood honey, sour, is rich in vitamin C as well. So, and I already mentioned about proline, which is found only in the bee saliva. And it must be present to show that the honey is pure. So just to summarize, we found that the different physical chemical properties of our honey were within the limit. And sourwood and tualang honey are the ones that we recommend because they have the highest concentration of phenolic, flavonoid, vitamin C. They are dark colored and they are probably the best among the 8 different types of honey that we investigated on. And then we also look at the mineral content of the honey using AAS, atomic absorption spectroscopy. And we found that sourwood honey has high sodium and potassium, both in a balanced amount, meaning that it's potentially good for hypertensive individuals because hypertensive individuals need to have both sodium and potassium in a balanced or high amount. And gelam honey has high calcium level, so is manuka honey. So we found the high sodium and potassium in the sourwood honey could be transferred from the leaves of this mineral because the leaves also have high sodium and potassium content. So we hypothesize that it is transferred to the nectar from the plant itself. Okay, and then we found that rubber tree honey is rich in iron, so it may be useful for anemic individuals. And sourwood honey is again has a lot of magnesium, and rubber tree has a lot of zinc, which may be useful for patients with prostate issue or acne. Yeah, so we found the different types of heavy metals or element mineral content in different type of honey. Although we didn't confirm that they are useful for the type of diseases that I mentioned, like hypertension, you should take sourwood or things like that. We didn't confirm. We didn't do research clinical trial on that, but we just found it in this type of honey and we recommend. That's it. We didn't go for the clinical trial. So this is my summary for the different type of mineral content. Manuka honey is rich in zinc and also calcium and may be useful for osteoporosis. Again, we didn't confirm. We just recommending it. Rubber tree honey is rich in iron and may be useful for anemic individuals. Zinc is also high in rubber tree honey and so on. Okay, then we proceeded on to look at the pesticide residue in the honey. And we investigated 10 different pesticides commonly used in Malaysia. So hopefully this one, we don't want them to be present. So good news, the honey we investigated, they were not contaminated with the pesticide. The reason this is important to be studied is because honey is an indicator for environmental pollution, because honey collected by the highway or the railway track contain high lead concentration because it absorbed from the environment. So this shows that honey should be collected deep into the jungle, far from pollution. So that's why honey is normally collected deep in the jungle by the orang asli, is it, Devi? They call it orang asli, right? The aboriginals.
Yeah.
Yeah. So overall, we found that the investigated Malaysian honey, they are pure, strong antioxidant with a lot of mineral present. But if let's say you want to use honey as a sweetener and you want to use it for cooking, do not put it in the hot wok when you cook because it will destroy all of the nutrient. So you shouldn't put honey into hot water as well. Warm water is okay, but not hot, not extremely hot. Okay, then we try to look at what are the volatile composition of honey. And from there, before we start, we found that methanol is the best organic solvent to extract the volatile active constituents because it pull out the most. However, methanol is not exactly a very green solvent, as you will see later on. And then we use GCMS to identify the volatile constituents, and we found 35. So this is a site study or a site track where, when we run the GCMS, we put the honey sample into the small 1.5 ml tube or the nano tube, which is made of plastic. While the honey is waiting to be analyzed for the injection, the petalate from the plastic sizer in the tube gets seeped into the honey. And because GCMS can detect down to nanogram per mil, we detected the plastic sizer in the honey. Therefore, here comes our another recommendation. Honey is best stored in glass bottle because if you store in plastic container, even for the short duration of the analysis of the volatile constituent, it already get transferred. And then when you consume the honey, you actually take the plastic sizer in. So, yeah, so that is another tips. Then we did on medicinal benefit, but because this topic is focusing on green pharmacy, I'm just going to show one medicinal benefit. So I want to inform you all that because we found that sourwood honey and tualang honey both have high antioxidant properties, we wanted to focus on this in the beginning of our study. So we wanted to focus on tualang honey, which is honey gathered from the tallest tree in the Asian rainforest. It's called Cumpacia excelsa. So a lot of bee farmers, they died because they fall down from the tree. It's a very tall tree. However, because this is a talk on green pharmacy again, it's not good to do research on tualang honey in our view because it has sustainability issue. We cannot get the resource easily now due to the rampant chopping down of tualang tree for development, for our house, for our building, and so on. And this is the reason why I switched to stingless bee honey now. But my initial study was to look at tualang honey and also sourwood honey. Anyway, this is a little bit of the data. We found that tualang honey has similar antibacterial property with manuka honey, but is even better for certain type of bacteria when compared to manuka honey. So this shows that our Asian honey is equally good or even better than the positive control, the manuka honey. And we also found that it's useful for wound infection and it can help to boost the immunity. In fact, during COVID time, a lot of people consume honey, right? Besides trying to get the sunlight. So honey actually help to improve the immune response of a person. So this is about talking about how do we go about doing sustainable research now. So we need to make sure that we engage with stakeholders as well, like the orang asli, the aboriginal, and so on. And so that after a long consultation, in the end, we decided to change from tualang honey to stingless bee because we can rear the bees. And it can be controlled in a controlled environment. So now I'm going to stingless bee. So stingless bee, we found that it has 3 times higher antioxidant. Do you know which one is the stingless bee honey? It's called kelulut, madu kelulut. Is it on the left or on the right? Anyone? Madu kelulut. Is it on the left or on the right? Just to have a little bit of interaction.
One, everyone. The right or the left? Still confused.
Stingless bee is which one? Anyone?
The right one?
Yeah, the right one is stingless bee honey. It's made in propolis pod. It's made in propolis, but the honeybee honey is made in wax, and wax have no activity whatsoever. It's just like it has zero nutritional composition even. So this is the reason why stingless bee has 3 times higher antioxidant, because the propolis itself has a lot of good properties. And because the honey sits in the propolis pod for stingless bee, it seeps into the honey, just like the just now I mentioned about the plastic sizer in the plastic container. It's the same thing. So this is the reason why stingless bee honey is better, beside the fact that it's more sustainable. And if you look at the bee size also, is how many times is that? Oh, sorry, it's 5 times bigger. The honeybee is 5 times bigger than the stingless bee. This is important because the stingless bee can go deep into the flower to get very good nectar, as opposed to the bee, the big bee, which has a fat body, if I can say, and they cannot go in. Yeah. So for this part of the study, okay, we actually have, beside honey, there are a lot of other constituents that are useful that comes from the bee, but I'm going to focus on propolis because to show about the green pharmacy effect. So propolis is a substance, is a bee glue, is the house of the, you can say the house of the bee, or and the bee also keep the honey in the propolis structure. It's the building material. And propolis, normally the farmers cannot use it because it cannot be processed. It's so waxy, dirty, and normally farmers will just throw propolis away, but it has a lot of medicinal properties. They didn't know. Even if they know, they cannot process it because it's very waxy. And it requires the addition of alcohol for it to be extracted. And when you add alcohol, it becomes not halal anymore, the product. So that is the issue that a lot of people or Muslim, they were experiencing. And I want to show that propolis has the caffeic acid phenyl ester that can improve memory and also has anti-cancer properties and also useful for multiple sclerosis. And it also increased telomere length and means that it keep your age longer. Your longevity is increased. That's why the bee farmers tend to live longer because we did a study on that, and their telomere length seems to be longer than non-beekeepers because they were constantly exposed to the bee sting and so on in daily doses. So it's good to be stung by the bee, no? So this is unless if you don't get allergic reaction, then it's good, yeah. So this is the product that we came out with. First, we use supercritical fluid extraction. It produces alcohol-free propolis extract, and it uses very safe solvent. And when you use supercritical fluid extraction, we can go low temperature. This is important for natural product design because it doesn't affect the flavonoid and phenolic acid content and preserve its stability. So we still have the antioxidant inside. And normally we don't go above 60 degrees. That's very high already for natural product because it will remove the antioxidant content. So you can see the different type of extraction method. So I circle the green ones, the green pharmacy ones, where water extraction is very, very good to the environment, or we can use microwave assisted extraction or ultrasonic assisted extraction beside the supercritical fluid extraction that I mentioned just now. So this is the solvent classification based on toxicity. And you can see that the recommended one, the less toxic, negative toxic there, is the water, ethanol, and so on. But as you go on, methanol and so on, it becomes more and more dangerous. So this is taken from Pratt et al. So stingless bee research for us is environmental friendly because it's sustainable. You can rear the bee at the back of your house or at the corridor. I have a bee. I have some hives. Okay. And
Really?
it is energy efficient. It uses green solvent. Sorry. And propolis, originally, the farmer threw them away. Now it can be used as health supplement because we managed to come up with the product, and the natural ingredient can be used for other downstream product like soap, shampoo, cosmetic. Oh, sorry, I'm going to. And this will also reduce reliance on synthetic compound. And then we also use recyclable packaging material like the glass, paper, rubber, which can be repurposed or reused after the product is finished. So our product won the Silver Medal Award for ITEX, and this was done in collaboration with the industry. So if I can recap the green pharmacy approach just now, we have, I think I finished early. We have targeted how many? Five? Sorry. Like preventing waste creation, using less hazardous chemical, using safer solvent, more energy efficient, and also using renewable raw materials. In fact, propolis is normally thrown away, but now it's like can be used. And this provide new job for the bee farmers, because now they can, instead of selling honey only, they now can sell propolis to researchers like us to be processed into a product. Seems like I finished earlier. So just to highlight on the challenges of green pharmacy. So a person or the researcher need to fork out a high initial investment cost. Like the SFE, just now I mentioned, supercritical fluid extraction, is not cheap. It's an expensive system. And normally we will share with other institutions, or we will use across different institutions, because it's too expensive for a university our university to purchase it. So we use somewhere else. And there is still a lack of standardized environmental regulation to date, also across the different world with many different regulation. So there is no one standardized one, as well as inadequate training and awareness among healthcare professionals and the public. And maybe this is the reason why it's important to do this type of conference, for us to be aware about green pharmacy and about using safe chemicals and not harming the environment over the long term. So I'm sorry, I seem to be finished faster than normal, because I was like, maybe I speak faster than normal. Anyway, thank you very much to Dewi and all the organizing committee for inviting me here. Thank you.
Okay.
We have more time for questions.
Okay. Thank you for Prof. Gan for your inspiring presentation. Let's continue to discussions, questions and answer sessions. Is there any questions in the box chat from audience? There's one from Norma Yasinta Wahdania. Allow me to read it. I am very interested in your explanation and presentations. I have questions regarding the contents of sourwood honey. Oh, this is the one of your material research, or honey in general, particularly concerning the presence of Clostridium botulinum spores that may be carried by honeybees. Could this contamination affect the beneficial properties of honey itself, especially its role as a natural antimicrobial agent? Maybe you, sorry, Prof. Gan, can you
Yeah, so
see the
the
chat
Nomi
book?
covered
Norma Yasinta.
Norma, yeah, she covered a very good point. Honey shouldn't be, actually, due to the spores, the presence of botulinum spores, honey shouldn't be consumed in children, and it should be abstained from children, because the spores are dangerous. And other than that, there is also the possibility of allergic reaction, which I touched a little bit just now. So normally, if you were to consume at least test a little bit by taking a little bit first, not the big dose. So the spores are not good. So that's why the bee farmers, they filter the honey first, because the spores are big size. So it will be hopefully filtered out. But if the bee farmer hit the honey, then it will destroy the constituent. So that's why normally honey is just filtered to maintain its properties. And then you mentioned about what is the best way to consume honey. So for me, I will just use 1 tablespoon and I will consume it directly. It's good for sore throat, especially if you have sore throat. And then if you can also dissolve in warm water if you feel that it's too sweet for you, if you consume 1 tablespoon like that. But just to add that, breast cancer patient who will be radiated because the area of the chest here is very close to the mouth. So the radiation can cause radiation mucositis. It's inflammation of the mucosa. So what the patient can do is to keep a mouthful of honey in their mouth when they are undergoing the radiation therapy. And then the honey will be heat up instead of their mucosa. And then after the process is finished, then the patient can either spit out the honey and maintain their mucosa, or they can swallow the honey also. So that is how honey can also be used during radiation therapy for breast cancer.
Yeah. Okay. Prof. Grant, usually we drink honey water
Yeah.
added by lemon, right?
Yeah.
Is it correct, or maybe there
No.
is, or there several negatively
I
effect of
Yeah,
that?
so because the lemon has vitamin C as well,
Mm-hmm.
they believe that it sort of like activate the honey to have a bigger effect. But I didn't really investigate but I don't think it will be harmful. It will be good to have both if you can. But whether it activates or not, we didn't do the
Oh
study.
yeah,
Yeah. So
because
it's a
and
good observation, Debbie.
because
Honey.
Indonesian people usually take that two kinds of material in the health drink, honey and
Yeah.
lemon.
Yeah, normally because when one has flu and fever, they want the high dose of vitamin C. So they add the lemon to increase the vitamin C content. Yeah.
Okay,
Yeah.
thank you. And next questions from Mrs. Enda
Yeah, I
Sulistiarini.
will Indah.
Okay, okay, Indah
I
Sulistiarini.
feel
It's
confused about how to identify honey that is truly of good criteria and beneficial for health. So the labeling criteria, I think you can ignore because most farmers, they want to sell their products. So they will say, this is pure honey, blah, blah, blah. The best one is to do lab tests, like how I did. But if you are by the roadside and you want to buy the
yeah,
honey by the roadside, there is one
it's more
method.
cheaper.
Yeah, there is one method you can do to determine whether the honey is pure or not. But you need a glass of water, see-through, and then you can take a tablespoon of honey, and then you drop it into the glass of water. And then if this is just by a crude observation, if it's pure honey, it should go in a swirls, and then it will drop to the bottom. But non-pure honey, it will, when you put it in the water, because it's sugar water, it will disperse. It will, yeah, it will change color almost immediately. But the pure honey, you need to
Sure.
kacau, you need to stir first.
Stir.
Yeah, then it will disperse. I hope that helps.
It's your experience.
Yeah. In
Okay,
addition,
your
many
practice.
honey products sold by local communities may have very limited lab tests
Yeah.
or quality evaluation. Is it still safe and beneficial to consume? Because I work with the industry, I will analyze the honey first, and once I found the company that produce a pure product, then I will only use that honey. Sorry. In Malaysia, we have this Mardi by Mardi. Mardi stands for what? MARDI, Mardi. It's the Malaysian Agricultural Research Development Institute and Pharma, I saw Pharma, Federation of Agriculture Malaysia
This is
Association.
government
Yeah,
institution.
so their tualang honey we did analyze and it found to be pure. So that one I can say because it's from government. But if I say about the honey from the industry, then I'm a bit biased here because I'm sharing that industry here.
Maybe in Indonesia we call it BPOM. Balai
Pepper?
Pemeriksaan Obat Makanan.
Yeah, okay. Yeah, so I mean, if you go for this type of agencies and organization, it's safer. Tend to be safer, yeah. But you can do that test first, the one that I mentioned. When you consume, when you buy the bottle of honey, you just do the test.
So you have to bring a bottle of water before you're going to buy honey.
Yes, it's very easy.
Yeah, thank you, Prof. Grant. Okay, to next questions from Ririn. Ririn Suarsanti.
In Indonesia, we call it klanceng. My question concerns the presentation of propolis. What percentage of ethanol is suitable for extracting it? This one depends on your machine and your other parameters. So I cannot answer it offhand because it varies depend on your experiment. So what is important is in my research, even we don't really use ethanol because it's not green. But if you do use, you need to make sure that you evaporate all of it off because it's dangerous to the body. So methanol can cause cancer even. Yeah, if it's still present in the product. So ethanol for halal is the percentage. I cannot remember the percentage of halal product for ethanol. I need
For
to check
drugs.
again. Yeah, there is a certain percentage for halal. It's okay to be present, but in small amount. Sorry, I forgot the percentage already. But in our case, we didn't use ethanol.
Okay. Ririn,
Ryan
is it okay
Radix.
for you? Or maybe you can continue with another questions directly?
Yeah, you mentioned that the presence of the presence of proline can be used as an indicator to determine whether honey is authentic or
The
adulterated.
next from
Could you
Mr. Ryan.
please explain how reliable proline analysis is? So amino acid, proline, it's in the, okay, so you are asking how do you determine proline content? Do you know I have already I need to check again. How do we determine the proline content in the is a type of amino acid? Anyone here can help? How do you determine amino acid present? What instrument is it supposed to be used? So because
Mr.
the
Ryan?
research is some time ago. Hold on, proline determination. Wow, your
Mr. Radix,
participant,
will
yeah,
you answer or maybe have directly discussions with Prof. Lan?
spectrophotometer. So it's measured by using a spectrophotometer. So the proline must be present minimum level, it has to be in the, you can look at for the International Honey Commission Guide 2002, and then you can find the exact value. So your your participant and committee member asked very good question. Very
Because you
good,
have
because I
a
cannot answer.
very amazing presentation, Prof. Gan.
Okay, using glass bottle for honey minimize the contamination of plastic sizer, and it's clearly more environmental friendly. How are glass packaging recycled? Because glass, oh dear, oh dear. How do you recycle glass? Um, I don't know the answer. How do you recycle glass? Susceptible to cracking and scratches. For us, we just using the visible, yeah, just the, but how do you know how many times? Like, you know, cooking oil, you shouldn't use it over 4 times, 4 to 5 times, right?
Yeah.
But glass, I'm not too sure. I will check.
Yeah, this is from questions from Mrs. Masito,
Yeah,
right?
very good question. I will, how many times can you recycle glass? No, I'm so sorry, I didn't check.
Maybe we can use that glass for another useful, maybe.
But
Okay.
glass, there's nothing that will leach into the content of the honey, right? So is even if it's broken, it's not really leaching into the honey. Let me, I'm going to get the answer soon. Wow, okay. So here it says that glass can be recycled an infinite number of times because it's made of natural resource like sand, soda ash, and limestone. So you don't need to worry about
Using glass.
yeah, using glass. So nice try, Masito.
Okay. Is there any questions from audience? Let me check to the YouTube. Is there any?
There is one
Okay.
more.
Okay. Oh,
How does
once
green
last questions,
pharmacy
Prof.
address the risk of carcinogenic compound like 5 during storage, especially in local honey product with limited lab testing? I can understand the pain actually, because in Malaysia it's quite similar to Indonesia. We are not that highly developed. That is why we just use the caution of consuming honey within 1 year of the shelf life, just to be on the safe side for 5 HMF. Actually, to be honest, not only honey, but so many other food, they are susceptible for risk of cancer, right? Even now, the environment is also so many risks from the environment, the pollution and so on. So the best we can do is just to go for quality food and like ensuring that the honey is of quality and to consume honey within 1 year. That's what we are practicing, because we don't know what will happen to the 5 HMF level after that, you know. So some people say all honey is good. I'm not too sure about that. Yeah. Hope
Okay.
it
Maybe
explained.
that's the last questions, Professor Gan.
Yeah. There is one more
Oh,
from
okay.
Madu Obat Shifa. I don't know what is this and about what.
Questions from Mrs. Eda or Afra?
I actually don't understand the question.
Mrs. Inda asked about the risk of carcinogenic compound.
That one is
It's
done.
already, okay. And then
Afra.
Afra Korean, I'm sorry.
Because it is in Bahasa Indonesia. I
Yeah,
cannot understand.
maybe you can answer in Malay.
No, I don't even understand the question in English.
Okay,
Sorry,
okay.
in
Uh,
the
in Quran, honey known as STIFAR or drugs, and the innovative
translate
opiol. It's
to
in
English.
Israel. Oh, I don't really get the point, but I try. In Israel, from the info from televisions, there's a bee ayu honey that produced without a bee. Is
I
that correct?
think this is, if the honey is produced without the bee, then it's not honey. So
Oh,
we
okay.
don't, yeah, we don't need to talk about how we apply green pharmacy into this when it's already contaminated product,
Okay.
isn't it? Yeah, it's not honey. If it's manufactured by human, it's not honey anymore. That's why the
Okay.
presence of proline must be there.
Yeah. Okay.
So
Okay.
I don't think we should confuse our mind further because this is not honey.
Yeah.
So
We have to make sure the content of the amino acid, right? The proline
yeah.
or the purity of the honey.
Very good.
Okay. I think that's all. Thank you for your question and answer, all participants, and the great answer from Professor Gan. Thank you very much. I have to end these sessions. To Professor Gan and all participants, as these sessions concludes, I apologize for any shortcoming in moderating these sessions. Thank you very much, and I will return it to the MC.
All right, once again, I would like to say thank you to Professor Dr. Gansiha, PhD, for such an insightful presentation. Now, we would like to present a certificate of appreciation to our second speaker, Professor Dr. Gansiha, PhD, for the insightful presentation and fellowship to this international conference. We would also like to kindly invite Mr. Najari Pharmacy Master of Science, PhD, to create and deliver the certificate virtually.
Okay, good morning. Thank you very much, Prof. Khan, for the wonderful talk and also discussion. On behalf of the organizing committee of the second SINACON 2026, we would like to express our sincere appreciation to our distinguished keynote speaker, Prof. Khan, for the insightful and inspiring presentation delivered today. Thank you once again for your support and participation in the second SINACON 2026. Thank you very much, Prof. Khan.
Yeah, thank you for the invitation, Bayu.
Yes, our
Bye.
pleasure, Prof. Thank you. Thank you,
Okay,
Prof.
bye-bye.
Yeah, we wish you all the best. Thank you, Prof. Bye-bye.
Mm.
Yep, thank you.
Ladies and gentlemen, now proceed to our third keynote speaker, Ayu Trimurti, Sarjana Farmasi, Master of Applied Science PhD from Sekolah Ilmu Farmasi Yayasan Farmasi Semarang. Ayu will present on the topic of next generation diagnostic enabled by advanced 2D nanomaterial for sustainable healthcare. Please welcome Apotheker Masita Suryaning Prahasiwi, Master of Science, as our moderator for this session. Mrs. Masita is one of the lecturer in Sekolah Tinggi Ilmu Farmasi Yayasan Farmasi Semarang.
For Mrs. Masita, the time is yours.
Okay, good day, everyone. Can you hear my voice clearly? Okay, I am Masito Suryani Prasi, who will act as the moderator for this keynote session. First, allow me to introduce our keynote speaker, Bayu Trimurti, PhD. Dr. Bayu currently serves as Vice Chairman of Student Affairs and Public Relations at STIFAR Yayasan Farmasi Semarang. He is also a principal investigator at the Nanotherapeutic Research Group, which focuses on integrating large-dimensional nanomaterials into advanced diagnostic devices and therapeutic agents. Dr. Bayu Trimurti received his PhD summa cum laude in 2023 from Taipei Medical University. And publication of Dr. Bayu is a quite long list. I will highlight some of the publications relevant to the topic today. For example, in 2025, Dr. Bayu authored an article in National Journal of Electrochemical Science published by Elsevier titled Electrochemical Nanobiosensors for Low-Cost Clinical Diagnosis. And in 2026, Dr. Bayu authored the article titled Deep Learning Assisted Piezoelectric Nanogenerators based on layered SNS2, MOS2 heterostructure for interactive sensing and effective computing application. It is published in Chemical Engineering Journal by Elsevier. Also in 2026, Dr. Bayu published an article in ACS Biomaterial Science and Engineering titled In Vitro Platforms in Bone Tissue Engineering from Biological Foundation to Advanced Models. Dr. Bayu's work strongly supports the SDGs of United Nations, especially SDG 3, 9, and 12. Now, please welcome Dr. Bayu.
Okay, good afternoon, everyone. First, I would like to check about my voice. Is it okay, the voice and also the slide? Okay, right. Thank you. Thank you, Bu Masita or Apotheker Masita for the nice introduction. This third session, I will convey several introduction and maybe like some of our recent studies about the nanomaterials. Because we are focusing on next generation diagnostics enabled by advanced 2D nanomaterials, especially for the application in sustainable healthcare. Well, before we are going through, I would like to like kind of check whether how many diseases that we have currently in the world, like globally. It is a very general questions to all participants. When we check in some of the paper or maybe recent reports that we have right now, like more than 10,000 type of the disease, yeah, currently roaming around our life. That when we divide it into two categories, like communicable disease agent and also non-communicable disease, then we can find, for example, like maybe in the past 3 or 4 years back, back in 2021, we have very serious and very detrimental outbreak, which is COVID-19. And this COVID-19 is shocking almost the world, right? Like everyone in the world are not quite ready with the outbreak. And also another type of virus like rhinovirus and even influenza is just very common, and it happens around us. And for the non-communicable disease, especially in Indonesia, we have cardiovascular disease as top 10 of the national disease that need to be resolved. And also we still have many problems in cancers as well, because for the cancers, currently there is no really effective cure to solve the problem. Yeah, that's why we still rely on radiotherapy, etc. And even when we are talking about the mental health, we still have many problems as well with the mental health, right? Even in Indonesia, currently the case is just growing because it has many reported about the mental health issues, etc. Yeah, well, this kind of disease, or a lot of and lots of the disease, how can we solve it, right? This is the big question. This is like a $1 million question. How do we combat those diseases? Like, okay, and then basically there are three ways, or there are three strategies to combat the disease. The first is the prevention. So this is kind of prevention in order to prevent the disease much more advanced before it spread. And then the second way to solve the disease is by the diagnosis. I mean, the development in terms of the diagnosis. And the last one is the treatment. So these three ways always comes together in order to solve the global disease. In our lab, in our particular lab, in which nanotheranostics research group lab, we are focusing on diagnosis. We try to combine between diagnosis and also treatment, but if we look at the percentage, maybe it's mostly about the diagnosis. And talking about the diagnosis, it's very related with one term in science, or in technology, or in recent technology, which is biosensors. So what is biosensors? Biosensors is an analytical devices incorporating a biological sensing element. So in general, there are two types of sensors. First is the biosensors, and the second one is the chemical sensors. When we talk about the biosensors, it should always incorporate this biological sensing element. This is according to the Prof. Anthony P.F. Turner, who is regarded as the father of the biosensors. And how about the main components of the sensors? So here we have four main components of the sensors. This is maybe more like an introductions to you all. The first part of the sensor is the target analyte. Maybe I will take a very simple example, which is glucometers, or the device that can be easily found in apotheke or in drugstore, which is glucometers, the simple device that can measure the blood glucose. So in this device, the analyte is glucose, okay? And then in order to measure the glucose need to be recognized by the biorecognition element. So then we come up with, to the second part of the biosensors. So we have the target analyte, and then we have also the biorecognition molecules. And how many types of biorecognition molecules currently have been discovered? And we have many types. Some types of the biomolecules that can be used as a biorecognition element. First, we have aptamer. So for the aptamer, this is kind of state-of-the-art technology, because aptamer has a DNA backbone as compared to the antibody. Many times aptamers was compared. Is compared to the antibody. In first way. For the aptamer, because it has the DNA background, the DNA backbone, so it can be synthesized in the lab. But for the antibody, it cannot be synthesized in the lab, because antibody we have to synthesize it inside the in vivo system. Inside the living body, for example, like rabbits, or maybe with the mice, mouse, etc. And then also we have protein and microorganisms, or even enzyme as the biorecognition molecules. So the main function of the biorecognition molecules is to recognize the analyte. Okay. When there is a binding event between biorecognition and also target analyte, there will be a change in physicochemical signals. Then the signal will be transmitted into the transducer part. Yeah, here The transducer's function is mainly to change or to convert the energy from the physical chemical energy normally into the electrical energy, and therefore it will be amplified into the measurable signal in the form of the analog signal, or maybe in the form of the digital signal. This is kind of simple. Okay. Yep. Yeah, this is like a simple illustration when the specific point recognition recognizes the analyte. But when there are another interference molecule, then of course it cannot bind to the biorecognition. And what are the key performance of the biosensors? Because when we develop a sensor, of course, we need to check, we need to ask whether is it good or not, right? And then what are the key performance? So the first for the key performance is the sensitivity. Yeah, now this is quite similar with when we develop an analytical device, or maybe when we check with the validation analysis, right? So first we need to make sure whether our device is sensitive, and then normally we will look at the LOD and also LOQ value, which is limit of detection and also limit of quantitation. And then the second one, we need to make sure whether our device is selective enough. How do we do that? We need to check whether we need to make kind of a design for the research to check this selectivity of the sensors. Normally, we will add a different type of the interference molecule into our system. Therefore, it can ensure whether our sensor can specifically detect our analyte. And then the third performance that we normally check is the measurement time. It's normally called by the response time. It's similar with the response time. So when we develop a sensor, we need to make sure whether our that our sensors can detect the analyte in a short time, not in a very long time, because sensors normally compare with the like instrument in lab clinics, because when we go for the like some checking in lab clinic in clinical lab, then it may takes quite long time, whether 1 or 2 days until we get the result. But for the sensors, normally the measurement time will be in seconds, or sorry, in minutes, or even in seconds. And then the fourth, we need to check the linear concentration range, the stability, and also the last, we need to also check the clinical applicability of the sensors. All right, and then what are the wish list of the biosensors devices? Okay, when we are talking about the ideal biosensor, or we call it by the point of care biosensor, it has to comply with the several parameters. The first, it need to be ultra sensitive, because the sensor need to has to be able to check, has to be able to measure the analyte as low as possible. Yeah, but it's not always as low as possible. As long as it complies with the clinical concentration, it will be fine. And then the second one is the highly specific parameters of the sensors. It relates with the selectivity of the sensors, and then it should be real time. So a good sensor should be real time. So it should detect the analyte in a real time manner, and then it should be high throughput, and it can be designed in a multiplexing way, because the modern sensors normally do not only detect a single biomolecules, but it can, within one device, it can detect different biomolecules or analytes. And then it should be label free. Yeah, because we don't want our sensors to be to be with label normally. Yeah. Yeah, and then it should be easy to use, because at the end of the day, the sensor has to be used by the patient. So we need to make sure before we develop the system that our sensors could be easy to use, especially by the user or the patient. And the next parameter is about the cost. So the sensor has to be cheap or not expensive. Yeah, it should be low cost. Yeah, because cost is a parameter that is quite sensitive to all of us, right? When we have a choice between one expensive and also cheap things, we normally choose the cheap things, and it's very normal. And then also we should confirm that whether the sensor is wearable or flexible or not. This is mostly related with the modern sensors. And then within many types of the transducers, for example, like optical transducer or maybe piezoelectric transducers, there is one particular transducers that has been widely developed at the moment. We have wrote a review article a few years ago. Then we check whether about the percentage of the electrochemical sensors and also optical, etc. And we found out that the most of the sensor that has been developed right now, mostly they rely on electrochemical system. Why? Because it's inspired from the success of the glucose sensors. Yeah, we can see here, this is the figure of the glucose sensor that we normally find in apotheke, right? In drugstores. We can buy it maybe around 300K or maybe for 400K rupiah. Yeah, it's quite cheap because it's already with a stick, sometimes 10 to 25 sticks. Yeah. And then this system is, I mean, the system use the electrochemical sensor system. I mean, the glucometer system depends on the electrochemical sensor system. So then we can have an idea, okay, this sensor, the electrochemical sensor has been commercialized. So why don't we use the similar concept with the electrochemical sensors, maybe towards another a direction, not only in glucose or in blood glucose. Okay, and then it's also simple. It's inexpensive because comparing with the measurement with, I mean, in the lab clinics, of course, the glucose sensor is much inexpensive. And then it can produce an accurate and also sensitive results, especially for patient diagnosis. And then it's easy to miniaturize into modern microelectronics, and it can be easily like upscale in especially in production stage. Here, I think just yesterday I found that how about the market value? Because I'm quite curious about the market value of the glucometers. Only one typical sensor, which is glucometer. And the market value for this device right now is 15.53 billion US dollar in 2025. And then it's projected to be double, around 30.18 billion in 2033. Maybe as kind of information for us, 15.53 billion US dollar equals to 17,3 triliun rupiah. I mean, the global market size for the, I mean, just for the glucose sensors. This is a kind of a starting point. It's just about the glucose sensor, and it occupies a lot of market size in economy. Yeah, but how about another disease? Because we have a lot of disease, right? Just like when we discussed in the beginning, cancers, heart disease, yeah, and even like what renal disease, yeah, and we still have many problems with the renal disease, etc. Okay, and with the growing trend of the nanotechnology, what can we do with this kind of technology and in conjunction with the electrochemical sensors? Okay, nanotechnology basically has been known from 1959 because the first the first doctor or professor that introduced the nanotechnology is Richard Feynman. This is very famous. In one of his lecture, he mentioned about there's a plenty of room at the bottom. At that time, people are still quite familiar. Not quite familiar with the nanotechnology. But then he has a sense. We had a sense that there are plenty of rooms at the bottom. I mean, with the many advantage within the nanotechnology. And then what is nanotechnology basically? This is a study of nanomaterials that include biological and non-biological structures. So for nanotechnology, it does not always associate with the inorganic materials, but it's also associated with the organic materials. Maybe as an example, we can see these simple figures. Like this is a nanotechnology that has been used in CNS disorder or central nervous system disorder, like Alzheimer's, etc. We have here the upside are the organic nanomaterials, which is nanoemulsion, for example, dendrimers, liposomes, and also polymeric nanoparticles, as well as pH-related nanoparticles. So all of these nanomaterials, I mean, the nanoparticles are based on the organic materials. And then on the other hand, at the bottom side, we also have inorganic nanomaterials, which is, for example, like gold nanoparticles. Right now, people are very eager to develop gold nanoparticle because it's very safe. Yeah, we don't always need to buy gold for saving, but it can be used also for research. Yeah, and it's very interesting because it has high biocompatibility. It's very compatible with our system, within our system. For example, in human system. And then we have also metallic nanoparticles, carbon tubes nanoparticles, nanocrystals, etc. And then we go further with the nanomaterials. When we are talking about nanomaterials, what are the actual size of these nanomaterials? It is debatable, to be honest. But then when I talk to people who are working a long time in nanomaterials, and when we check in several papers, several literatures, we can find that it refers mostly for the organic materials. It refers mostly to the specific scale or range within 1 to 100 nanometers. So when we design the nanomaterials, ideally it should be below than 100 nanometers. But then when we read another references, we can find as well that the nanomaterials can be the range can be just below 1 micrometer or 1 micron. So as long as it's below or less than 1 micrometer, it can be called by the nanomaterial. So it's still okay to have the nanomaterials just below 1 micrometers. Okay, between several types of nanomaterials in our lab, we are focusing in one particular type of nanomaterials, which is called by 2D or two-dimensional nanomaterials. Yeah, we have 4 types of nanomaterials, right? Like 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional. In a simple analogy, we can see these 2D nanomaterials just like a piece of paper, just like this one. Okay, when we talk about a piece of paper, it has a very large surface area, but when we check the thickness, it's very thin, right? Just like this paper, it's very thin, but it has very large surface area. Just imagine this kind of nanomaterials can be kind of like a bus cargo, right? To bring our drugs, for example, compounds into the receptor, because these nanomaterials can be used as, in a drug delivery system. Okay, just to help the drugs to achieve the receptor more efficiently and also more effectively. So why we are interested into the nanomaterials? Yeah, so that's one of the reason, and another reason is because it has high electrical conductivity and it has also fast electron transfer kinetics, and it's also very easy. I mean, not very easy, but it's quite easy to be functionalized. Okay, and also we are here focusing on one particular type of 2D nanomaterials, which is tin disulfide. What is tin disulfide? Basically, it's an inorganic graphene analogs. So maybe we are very familiar with the graphene. Yeah, graphene with the honeycomb structure. Just like before, Prof. Khan discussed a lot about honey, but graphene, it has a honeycomb structure because it's very stable. And then for the tin disulfide, it's quite similar. I mean, the structure with the graphene. Okay, but the difference is because it's an N-type semiconductor with a wide band gap. Okay, and then it has good stability as well in acid and neutral aqueous solution, and it's widely distributed. I mean, in our planet. So therefore, the price is quite cheap. I mean, to buy these materials, and also it has lower environmental toxicity. So this is well aligned with the main topic of SINACON today. Okay. Yeah, and before we move forward to this to our study, first we would like to introduce about the self-powered device for biomedical application. I firstly known this technology when I was in Taiwan for PhD study. My professor's expertise is basically on self-powered device, and at that time we are interested to develop a sensors. And then we think when we discuss, why don't we combine each other between biosensor and self-powered device? Okay, then it come up with an idea to be a self-powered biosensor system. What is self-powered device? So just imagine about our cell phone, right? All of us has the cell phone or handphone. And every time when the battery is off, I mean it's almost off, we have to charge the cell phone. Okay, so that's the normal way how we deal with the cell phone. But then for the self-powered device, the device itself can power itself, so we don't need the battery in order for the cell phone to be function. Okay, and how does it happens? How can it happens? Like basically the working mechanism, we deal with the TENG or triboelectric nanogenerator. So there is a particular system here. For example, we have tin disulfide and we have eco-friendly substrate at the top and also at the bottom side, and then it aligns each other. And also when it contact and also separates each other, it will generate enough power or enough electricity, I mean, to be used to function the device. So this is very interesting as the future direction of the sensor, because we have kind of a belief that maybe in the future the biosensors or even the medical devices, we are not we're not going to really rely on the battery or the charging system. Yeah. So hopefully in the next few years, then we can truly realize this idea. Okay, and now I come up with one particular example of what we have developed recently. So we have a study about the 2D SNS2 based biosensor for amyloid beta detection in Alzheimer's. So this is basically my PhD study. Yeah, at the time, because it was in Taiwan. So Taiwan has a problem with Alzheimer's, or Indonesia we call it by pikun. Right? Maybe in our country, we have different type of the disease, like for to be resolved. Right? Yeah, we don't have many percentage for the pikun in Indonesia or Alzheimer's, but in Taiwan they have a lot of problem with the Alzheimer's. And one biomarkers that can be used to as a indicator for the Alzheimer's is amyloid beta protein. Okay, at that time we developed the tin disulfide nanomaterials, and then in order to be embedded with the biosensor to detect this particular protein. And the study has been published in Chemical Engineering Journal, one of the top journal, I mean, in chemical engineering area. Okay. And then the first question, how did we create the tiny nano SNS2? We use the simple synthesis approach for developing the SNS2 or to grow the SNS2. Here, we choose the dry system, I mean the dry approach of this synthesis. We didn't use the wet synthesis at the time. So we have two different precursors, which is the sulfur and also tin chloride pentahydrate. When it's vaporized inside the tube, because this is the kind of illustration for the tube. Here we have very high degree of the temperature. We heat it up to 40-50 degrees Celsius, and the sulfur will be vaporized. When the vapor of the sulfur and the vapor of the tin chloride meet each other, they will kind of carry out a simple reaction that can form SNS2 or a tin disulfide. Yeah, like here we have the tin disulfide. Like this is the illustration, and right at the left side, basically is the SEM image of the SNS2 after we successfully grow the materials. Okay, and then before we are checking the biosensor system or to check the analytical parameters of the biosensor, we have to design like the chemical functionalization onto the top of the SNS2. So at that time we use the MPTMS. Yeah, MPTMS is the specific kind of organic molecules that can harvest a lot of thiol groups. Okay, here we have SH, this is the thiol groups. But then it has also some disadvantage because the thiol group, when we leave it outside, it will bind each other to form the disulfide bond. So this is the disulfide bond that can block the system. Okay, if it's blocked, then we have to cut it, right? We have to cut it by the specific biomolecules. Then we use the specific another molecules which called by DTT. So DTT is the thiotryptol. We use it to cut the disulfide bond, and then it will form again the free thiol groups here. And then these thiol groups will afterwards we add it with the 5N thiol aptamer. So this is the aptamer, I mean the biorecognition molecule that we use. Okay, and then when it attach onto the surface of the system, then it can bind each other with the formation of the disulfide bond. Okay, afterwards we add the BSA or bovine serum albumin in order to block the in order to prevent the non-specific binding. And then finally, well, we can use the complete biosensor system to detect the amyloid beta protein. Okay. Yeah, this colorful figure, this is the representation of the protein, I mean amyloid beta. It's kind of mechanism, like electrochemical mechanism for the system. So we explain it in our paper that we rely on the redox reaction, reduction and also oxidation in order to measure the amyloid beta 42 biomarkers. And then the first result is about the nanomaterials characterization. The first things that we did is we measure with, I mean, the SEM within the SEM or scanning electron microscopy. We have to prove, right? We have to prove that, okay, you claim that this SNS2 has been formed, but how can you prove it? Okay, and then we use the SEM image. Okay, and then within the system, we can have the very highly distributed SNS2 onto the substrate. So when we enlarge it like around 200 nanometer scale, then we can see here, this is many SNS2 has been grown in our substrate. Okay, and then the next we measure with the Raman scattering. So we need to make sure whether this is the true SNS2 or maybe another form of the nanomaterials, which is SNS or etc. And then, okay, then it has a specific A1G type of the signals that is very specific for the SNS2. And then we move forward with the PL or photoluminescence measurement, and we compare at the time the bare substrate without SNS2 and also the SNS2 with the substrate. This is the direct comparison with the control. Normally we call it by the control, and we can see here the very distinctive signal appear. Yeah, when after we synthesize the SNS2, and also we carry out the XRD measurement as well to check about the crystal structure of the SNS2. And then further, we prove the nanomaterials with the TEM system. Here we use the high resolution TEM. And here we can see the atomic arrangement of the SNS2 and with the D spacing around 0.319 nanometers. Okay, and for the figure D, E, and F, this is a kind of a proof or maybe evidence that the thin element or SN element has been well distributed into the single nanoseed of the materials, also with the sulfur elements. Okay, and then once we successfully develop the SNS2, we use this SNS2 into the biosensor system. So this is a kind of analytical parameters for the biosensor system. For the figure A to D, basically we check the linearity and also sensitivity. We improve or we increase the concentration of the biosensors, the amyloid beta. Yeah, and then here we can see that it's very easy to be understood. It's more or less like like a curva Baku, right? Or maybe a standard curve. Okay, when we increase the concentration, we will see the increasing response as well of the system. We use the electrochemical system over here. And then here we can find the determination coefficient is very close to 0.99, because when we look at the coefficient of the correlation, it will be 0.99. Okay, and then for the figure A and B, we measure the system under the PBS, or forced part buffer saline, and then afterwards we check the system inside the human serum, because at the end of the day, we hope that our system can be used to detect the amyloid beta in a real human serum. Okay, and then for the figure E, this is basically the figure for this selectivity study. Okay, we employ different types of the interference molecules, like amyloid beta 40 monomers, fibril, oligomer, and even dopamine as well, and some salt, including NaCl and so KCl, and also the glucose, in order to confirm whether our system really works to detect the amyloid beta or not. And as a result, we can see here that the system specifically measure the amyloid beta of 42 oligomers. But then when we check the different biomolecules, it will result quite low, low response as compared to the amyloid beta 42 oligomers. Yeah, it has been also tested statistically. Okay, and for the figure F, we use the stability study, or we carried out the stability study in order to check whether how stable our sensor is. So then we prove that our sensor are stable within the 30 days of the storage. And I still remember at that time we received many suggestions from the reviewers during the peer review process of our papers. That one of the reviewer raised question about how about the real blood serum test result of the sensors, because we didn't include it before when we submit the paper. And then afterwards we collaborate with a medical doctor from Chang'an University in Taiwan. And then our collaborator were able to develop the APP knock-in transgenic mice. So we collaborate in order to have the mice model with Alzheimer's. So it's very challenging to develop mice model with Alzheimer's, because normally for the people to have the Alzheimer's, normally the age should be over 65 years. But then just imagine how about in mice? It's not possible for us. It's impossible to wait many years for the research, right? For the normal research, we hope that can be done in 5 to 6 months or even in 1 year. But for the Alzheimer's, it will take a longer time. But then there was a solution at that time, because we used the APP type of knock-in transgenic mice. Then we were able to develop Alzheimer's animal model. And then we withdraw the blood, and also we extracted the serum, and we tested our sensor to the serum of the mice blood. And then as a result, we can see here for the figure A that for the biosensor response, we compared between normal mice and also AD mice. For the AD mice, the response is higher. And then when we convert it into the concentration, then it will be statistically significant. I mean, the difference, okay, between Alzheimer's and also normal mice. And further, we also analyze the dissociation constant of the aptamer and also AB42 complex. So we use the Langmuir isotope model at that time, and we were able to calculate the KD, or the dissociation constant, to be 1.57 nanogram per mil, and also 0.3 nanogram per mil for under the human serum system. So what are the conclusions this first study? So first, we a label-free, cost-effective, and highly sensitive biosensor immobilizing amyloid beta 42 aptamer onto functionalized disulfide has been successfully developed at the time to detect the AB42 oligomers. And then we utilize the vertically aligned nano SNS2 with the facile CVD system, because in our synthesis approach, we use the chemical vapor deposition, or we call it by CVD system. And then for the limit of detection in physiological buffer and in human serum, we found it very interesting because it can be down to femtomolar level of or FM. Normally, when we for example make a solution, normally we make a solution like right up to nanomolar level. Okay, molar, millimolar, micromolar, and nanomolar, right? But then the sensors are able to detect the amyloid beta 42 in a femtomolar level, so it's much below than nanomolar level. Okay, and also the sensor was used to determine the amyloid beta oligomers in mice blood sample, and the SNS2 APTA sensor has a noticeable selectivity, stability, and very low KD. So it shows a prominent binding affinity between the aptamer and receptor and also the analyte. And also the proposed biosensor design can shed light on the future of Alzheimer's diagnosis. So this is kind of the first study. Maybe due to the time limitation, I will jump. We have 3 here, but then I will share the last one. Yeah, and then once we return to Indonesia, what did we develop? Okay, so we are now developing a different type of sensors because we realize the facilities of the research is quite different between Indonesia and Taiwan. So we try to adjust the sensors, and we were developing right now the paper-based sensors. Okay. For the paper-based sensor, it belongs to the low-cost sensors because we have different types of the sensor here, like filter paper, glossy paper, etc. And we here use the micropad sensors. And also for the fabrication approach, we use the screen printing and normal printing approach, which is very easy to print the paper, okay, to be developed as the sensor system. And then for the sensing technique, we normally here we develop or we use it we use colorimetry approach because we currently we didn't have the potentiometry and voltammetry to be used. Okay, and for the biomolecule sense, we have developed sensors to detect the dopamine and also glucose, and also glucose with the creatinine. So this is what we are developing at the moment with our undergraduate students. So we develop the sensors, I mean the paper-based sensor to detect dopamine as early as possible, because I mean this is more and less related to the CNS system. Right now we have many things about mental health, right? In Indonesia, many cases of the mental health, and one of the particular biomarker that can be used to as an indicator for the CNS disease is dopamine. So we develop the sensors that specifically detect the dopamine. So the sensor has been completed, and now we are in the stage of writing the manuscript. And also we developed another paper-based sensors to detect the glucose because we still see many challenges with the current glucometers, like the use of the blood. Okay, people are not sometimes, for some people, not all people, some people they are quite afraid to see the blood, and then it will be a challenge. It will be then an opportunity to ask how do we develop a sensors that did not use the blood as the sample, but we use it like a sweat. Okay, sweat or maybe urine. Okay, for the urine, it's non-invasive approach. Okay, just not like blood, because for the blood we should take it from our IV system, but then for the urine we don't need to use an invasive method to withdraw it. Okay, and then also we develop the glucose and creatinine based sensors to detect the particular disease which is called by the diabetic nephropathy, because in Indonesia we have increasing evidence as well of the diabetic nephropathy. This is basically comes from our discussion with one of medical doctor, a specialized medical doctor in diabetic nephropathy or in renal system. Okay. And then these 3 types of sensors that have been developed, and hopefully in the future we can develop different types of paper-based sensors as well. So now we come up to take-home message. The first for our take-home message from today's session is 2D nanomaterials enabled by sensor are revolutionizing early disease diagnostic through ultra-sensitive, rapid, and label-free detection technologies. And also, we were able to develop as well the flexible and portable and sustainable sensing platform that can pave the way for accessible point of care medical device. And what about the future direction? Okay, the future direction of the sensors could be an AI-based self-powered wearable device, and also the greener approach for the for developing nano biosensors, just in line with our main topic of the SINACON today. And then the last one, for the next generation healthcare technologies, strongly support the SDG 3, 12, and also 17. So I here strongly encourage that all of the researchers in Indonesia, or maybe all over the world, that we need to think that what can we contribute to the SDGs from now on, I mean, in terms of our research. Yeah, it's very nice. It's very important for us to always align our research purpose with the SDG, Sustainable Development Goals. So I would like to acknowledge and thanks to our collaborator in Taiwan. This is my previous advisor, professors in Taiwan, Prof. Yang, currently associate professor in NCU, and then also Dr. Huang, my mentor at that time because he was postdoc, and now he's in USA. And then all other lab members. So the up figures is when we just finished from a dinner. So that's why we look happy here, because we just finished from a dinner. When we came back to the lab, then it will be a different story, right? Okay. And then we also, thanks to our research group member, Dr. Atika, as an like co-PI in our research group. And also we have several students that have graduated from our lab, because it's just established last year. Iza, Mazaya, Anindya, Khairunisa, Farha, Royhana, Brinanda, Devita, Hanif, and also Tabitha. Thanks for the hard work, and hopefully we can continue the fruitful collaborations in the future. So if you are interested in our research group, you can easily scan this barcode, and then we can collaborate anytime and anywhere. So my final message is, let's collaborate to advance nanotechnology-driven sustainable healthcare in our country, Indonesia. So thank you. Thank you very much, and I will get back to the moderator. Thank you.
Thank you, Dr. Bayu, for the presentation. It's really interesting because we might be more familiar with nanotechnology in pharmaceutical preparation, but this one is about diagnostics. Okay, we have a few questions already. First question is from Mrs. Atika. She'd like to ask, can we combine traditional herbal medicine with the nanobiosensor technology? And she saw there are some computational findings in your presentation. Is it possible to run simulation on that? And how about charge transfer? Can we do the simulation as well? And is it possible that sensor signal will be shielded by the ions in high soil environment like human blood?
Okay, thank you, Dr. Atika. It's very tough question. Well, actually, it has maybe 3 or 4 questions, right? Okay, the first question: can we combine the traditional herbal medicines with the nanobiosensor technology? The answer is yes, definitely yes. Right now, actually, there is one particular study that I have not able to share today, and it's about the sensor for the antioxidant. Yeah, so we developed a simple pepper-based sensors to detect the antioxidant compound, I mean, within the herbal sample, etc. So it will be kind of a breakthrough. Actually, it's not new because it has been developed a long time to our also bachelor supervisor when we were still in University of Jember. They develop many technologies in antioxidant I mean sensors. Okay, we can develop like a pepper-based sensors to detect the antioxidant compound. Yeah, and because it will be kind of alternative. Right now, when we like a normal study, when we want to know whether this herbal or this sample has kind of potential in antioxidant or not, we use the instrument, right? Like spectrophotometry, UV visible, etc. But then why? And I mean it's based on color change. Okay, why don't we bring this concept into the biosensor system? Because in paper we can easily see whether the color change quickly or not, like from the purple to yellow, for example. So yes, it's possible. And also, the second question will be also there are some computational findings in your presentation. Is it possible to run the simulation on that? Yes, the answer is yes. Because actually most of our study, it consists of one computational approach, which is called by DFT or density functional theory. So the first keynote speaker today, Dr. Faiz, explained many things about the molecular docking, right? But then in material system, we normally use the DFT system to run to explore the electronic properties of the materials, etc. So we use the one particular techniques, which is called by density functional theory, and it can be easily found. I mean, the tutorials in Google. Okay, and then how about the charge transfer? Can we do the simulation as well? We were able to detect the charge transfer by measuring the potential energy. I mean, yeah, we have done several simulation on it, and it has been published, I think last year, 2025, in Nano Energy Journal. We simulate the tin disulfide, and as I remember, it's in Ecoflex polymer, and then we observe the potential energy on it. And then is it possible that the sensor will be shielded by the ions? Yes, it's very much possible. Yeah, because in blood, as we know, blood system does not contain only a single metabolite. It has many things like salt and proteins and even the cells, like blood cells, etc. So it can be. I mean, it's possible to for the sensors to be shielded. But then we have also one particular strategy to avoid this kind of problem. Yeah. Yeah, that's the first question. I mean, the answer for the first question, Mrs. Masito. Yeah.
Okay. The second question is from Mr. Sorry, pardon. The second question is from Mr. Ian. He would like to ask how biosensor technology can be applied for the identification of secondary metabolites from medicinal plants. Could you explain the general workflow?
Yes, thank you. Thank you, Dr. Ian, for the very good question, right? Is it possible for us to develop the biosensor technology that can be applied to identify the secondary metabolites? Yes, the answer is yes, it's possible, as long as we can apply the basic concept of the sensors. So for the secondary metabolites, if there are some, for example, biorecognition molecules like enzymes or antibody or aptamer that can detect these metabolites, then it's possible to develop the biosensor system within this idea. Okay, so it's very much possible. But I will be very honest, because I'm not quite familiar with the herbal medicine. Maybe it is kind of opportunity for us to collaborate in the future if we want to develop sensor to detect the secondary metabolites. Yeah, that's my answer.
Okay, and the third question is from Mrs. Unsari. She would like to know about the APP knock-in mouse model. After being induced, what responses are measured to confirm that the mice are positive for Alzheimer's?
Yeah, thank you, Mr. Unsari. Yeah, oh, sorry, Mrs. Unsari, right? Thank you, Mrs. Unsari. This is like all the way from Queen's University Belfast. So hopefully you're always great there, Mrs. Unsari. And for the question about the APP knock-in mouse model, after being induced, what response are measured to confirm the mice are positive for Alzheimer's? To be very honest, at that time, because we have very short time, this particular study of our, this particular study when I was PhD at that time, this is from the one of reviewer suggestion. So we have very limited time. So we have, I remember still we have only 1 month to prove whether the sensor has, can be used to detect the AB42 in a blood of Alzheimer's mouse model. And because it's very limited, then we ask that our collaborator, we request our collaborators in different university in Changgang University to kind of provide us about this APP knock-in mouse model. And I'm not in this field, unfortunately. I'm mostly working with the nanomaterials and then the analytical devices, etc. But as I remember, what they use to confirm whether the mice are positive for Alzheimer's, they use the behavioral approach. So they first confirm with the whether they have many amyloid beta 42 inside the brain system. So normally they will use the some specific fluids from the backbone, and then afterwards they check the mice with the behavioral test. Correct. Just imagine when people have Alzheimer's, because they forget almost everything. So sometimes, many times, they are not able to walk normally, right? For example, within one distance to another distance, like 10-meter distance for the normal people, it can be easily for us to walk. But then for the Alzheimer's people, maybe they will be confused. So there will be a delay in time. So then these things was brought into the lab. Yeah. So the mouse normally measured by these techniques. Maybe we can have a chit chat after this about how to measure this question, right? Thank you, Bung Sari, and you can also Google it. It's to easily find the also to confirm the answer. Thank you.
Okay, there's quite a lot of questions already, but I will choose some interesting one because our time is sadly limited. So either is Mrs. or Miss Nur Habibah from Poltek Surakarta wants to know how to deal with the matrix in biological sample if we use biosensor.
Okay.
What are the critical points that must be considered when developing a biosensor?
Yeah. What are the critical points that must be considered when developing a sensors? The question will be the biorecognition element. So before we develop a sensor, we have to make sure the biorecognition element that we are going to use is the most proper one. Okay, like for example, for the glucose sensor, it will be effective if we use the glucose oxidase enzyme, because the glucose oxidase enzyme can oxidize the glucose to be a gluconolectin, and it will also result like a color change. So it will be quite easy to observe these changes. I mean, to use for example, the colorimetry approach for this sensor. So that's my question. I mean, that's my answer. The most critical points is the choose of this selection of the biorecognition element. We need to carefully choose whether we are going to use enzyme, aptamer, or antibody, etc. Yeah. Thank you, Mrs. Nur Habibah. Yeah.
Okay. Dr. Bayu has answered for question. There are so many people are curious about this topic with Dr. Bayu, but sadly our time is limited. Regularly, we don't have time for all the question. But if you want to ask Dr. Bayu further, you can contact him via email displayed on the screen.
Thank you. Thank you.
Thank you very much, Dr. Bayu, for the presentation and the Q&A session. And so this is the end of the third keynote speech. And I will return the floor to the MC. Thank you.
All right, Mr. Bayu, thank you for such a wonderful presentation. Now, we would like to present a certificate of appreciation to our third speaker, Mr. Bayu Trimurti, Sarjana Pharmacy, Master of Applied Science, PhD, for the insightful presentation and valuable contribution to the international conference. We would also like to kindly invite Dr. Apoteker Endang Dwi Wulansari, Magister Science, to join us on screen and deliver the certificate virtually.
Okay, on behalf of the organizing committee of the second SINACON 2026, we would like to express our sincere appreciations to our distinguished keynote speakers, Mr. Bayu Trimurti, PhD, for the insightful and inspiring presentations delivered today. As a token of our gratitude, we are pleased to present this certificate of appreciation virtually. Thank you once again for your support and participation in the second SINACON 2026. Thank you very much, Mr. Bayu.
Okay, thank you. Thank you, Dr. Devi. Thank you, everyone. Hopefully you enjoy the talk. Thank you.
Ladies and gentlemen, we now proceed to our fourth keynote speaker, Professor Wafa Al-Jabal, PhD from School of Pharmacy, Queen's University, Belfast, Northern Ireland. Professor Wafa will present on the topic of nanomedicine and advanced drug delivery, transforming pharmacology and therapeutics. Please welcome Mrs. Atika Darumas Putri, Sarjana Pharmacy, Master of Applied Science, PhD, as our moderator for this section. For Mrs. Atika, the time is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh, and good afternoon, everyone. So my voice still sounds clear, right? Okay, and very warm welcome back to this session for the participants. I do hope you've managed to grab a quick coffee or a bit of tea, yeah, previously. And Assalamualaikum, Prof. Wafa.
Waalaikumsalam. Hello.
Yeah, hello. So it's really good morning there, Prof. Yeah,
Thank you
and
so
thank
much.
you so much. Yeah, thank you so much for your acceptation on our invitation previously. And it's truly been a pleasure for us to meet you here, even if it's virtual. But I believe that I and all the participants here will learn a lot from you. Yeah.
Thank you very much.
Okay. Yeah. Prof. Wafa, before we really begin, I would like to introduce you first to all our participants. Let me share the screen. Think it's clear enough, right? Yeah. Okay, so Professor Wafa Jamal is a professor of advanced nanotherapeutics at Queen's University, Belfast, UK, and an international recognized leader in nanomedicine and translational cancer therapeutics. Prof. Wafa is also a Perseus and UK registered pharmacist. She completed her PhD in drug delivery and nanomedicine in 2008 from the School of Pharmacy, University of London. We may now know that it's, I think the university changed its name to UCL, yeah, University College London at School of Pharmacy. And her research focuses on the design, development, and clinical translation of advanced nanomaterial-based therapies, including molecularly imprinted polymers or nano-MIPs, mRNA delivery systems, and multifunctional nanoparticle platforms for cancer immunotherapy and theranostics. During her career, she, of course, has published lots of high-impact journals, yeah, such as recently in Elsevier, International Journal of Pharmaceutics, ACS, lots of published papers in Nature's Royal Society of Chemistry as well. Yeah. She is currently recognized for her contributions to the field, as she has been listed among the Stanford top 2% scientists worldwide in 2020 to 2023. She is an active member of the international research community as well, serving on editorial boards, funding panels, and scientific advisory committees, also delivering numerous invited keynote and plenary lectures like today's talk. Yeah. In addition to her research leadership, Professor Wafa is committed to advancing education, equality, and interdisciplinary collaboration as well. All right, before I really hand the session over to Prof. Wafa, a quick reminder to all of the audience here that please do post your question in the chat box throughout the presentation, and we will address as many as possible at the end. Okay, without further ado, it is my honor to invite Prof. Wafa to take the principal floor. Over to you, Prof.
Thank you very much, Atika, for the nice introduction, and it's a great pleasure to be here. As you mentioned, it's a bit early here to me, so hopefully I'm fully awake for this presentation. So let me just share the slides first. And okay, I have to say I'm not a big user of Zoom, so I'll try just to find the document. Okay, so share. Sorry. So can you see my slides?
I don't think, maybe, is there any issue in the side, or should I help you?
So currently you don't see them?
Yeah, not yet.
Okay, so let me just go back. There's too many windows open, so I don't know. So, okay, so the share button, is it?
Yeah, the one with the arrow sign.
Well, it shows me share, but then
Yeah.
share entire screen. Okay, file. Yeah, I can see the file. Okay, so documents. Okay. Okay, so
is there any share button, Prof, in your
yeah,
Zoom?
I can see it. I'm not sure if, to be honest, if it I see the share button, but I'm not sure if it's security doesn't allow me to open the. Okay,
Is there any notif on that notification that pops up? Oh.
Well, it says share screen. It gives me, it doesn't allow me just to attach the file. I see the shared button, but it doesn't allow me to. Okay, I'm a bit stuck here.
Yeah. Shall we, yeah, maybe is that if it's possible for you, I may help to run your PowerPoint slides.
Well,
That's possible.
well, I have to send it to you because I didn't have it.
Sure. Okay, bro. Yeah. You may send me in private message in the chat box.
Okay, the problem is the file is a bit big. That's why I'm
Okay.
let me just try.
Or maybe via email also fine, Prof.
Okay, 1 second. I'll send it maybe by email.
Okay.
Bu Arik, emailnya.
It's just taking a bit of time because the file is big, so.
You can send it to SINACON email, Prof, that previously contacted you.
Yeah, I'm just uploading it to the OneDrive because it's too big for the attachment.
Okay.
Okay, it doesn't allow me. Yeah, I just sent the link if you can open it, or just let me know if you cannot access the file.
Okay, let me check. Yeah. Okay, I got the file profile. I'll try to download it first.
Thank you.
This is really huge file profile.
I know. That's why I couldn't send it before.
Yeah,
Yeah.
yeah. Okay, let me try to finish to download it first.
And first it seems.
Prof. Wafa?
Yeah.
Yeah, now I'm able to download it already. So I will share the screen for you. Is that all right?
Excellent. Yeah, thank you very
Okay.
much.
Okay. Yeah. Okay. Okay. So this the shared screen already appeared, I think. Let's try to play this.
Okay. Are you sharing the
Okay.
slides? So I don't
Yeah,
see
yeah.
anything.
Can you say not yet? So how about now? I'll try to run it the slides. So are you able to see the slide now, bro? Or not yet?
No.
No? Okay.
I don't know if it's my screen, to be honest, or I don't know if the audience can see it, but
actually, yeah, some of the committees here are able to see that, but let me try it again. Okay. Yeah. Rob, how about now?
no, I still.
Yeah.
Let me just, I'm not sure to be, I'll just exit this.
So,
Hello?
yeah.
Yeah, I can see it now.
Oh, you know can see it. Okay.
Yeah, finally,
Maybe,
yeah.
yeah, maybe better to show it like this. Because if I arrange it to PowerPoint present a slideshow, I think that
Yeah.
will make a different burden for the connection, for internet connection. Is that okay if I put it this way?
Well, I'm not sure if that, because I have some animations. So
Oh, I see.
would I
Okay,
have
maybe.
control? Would I have control of the?
Oh, I don't think, but that's okay. I'm trying to show it like in right now in a slideshow version. Can
Yeah,
you
but
see
will,
it?
yeah, but will I have control of the slides or?
I think I would do it for you. Yeah, because maybe you can just say next and so on. Change
Okay,
or
okay.
that, yeah. Okay.
All right,
that's
okay. Sorry for the delay. I mean, it's a
No
technical thing.
problem, no problem. Yeah, it's okay.
Okay, thank you
Yeah.
very much for the invitation and everyone for being patient with us. So it's a great pleasure to be here today and tell you more about our work in the field of nanomedicine and how this could transform pharmacology and therapeutics. Next, please. Next. So these days we have a fundamental shift in the way how we think about medicines, going from the not only thinking about how we deliver, what type of drugs we deliver, but how and where and when we can deliver them. And nanomedicine have been at the center of this transformation. Traditional pharmacology has been limited with the tissue biodistribution, systemic toxicity, and all the side effects associated with suboptimal efficacy. Using nanocarriers, as we see here on the left-hand side, we can see these nanocarriers, very small, tiny particles that 60 to 80 times smaller or thinner than a human hair. They could be fabricated using different type of materials to deliver, to encapsulate cargoes and therapeutics, navigate through the body, protect them from degradation, and reduce toxicity. The particles could be designed in a way that they are more targeted. They can go to specific tissues in the body, and they have the capability as well to deliver the drug upon on-demand drug release. And selectively with the combination and their capability to cross biological barriers, that would result in specific and high selective accumulation in tissues combined with on-demand drug release that ultimately increase the localized concentration and reduce side effects, and that improve the patient outcome. Next slide, please. So looking at this slide, this is echo that transformation I mentioned about nanomedicine, and that gives you, it's a very busy slide, but just to give you the diversity of the nanomedicine that have been used in terms of composition, shape, the different therapeutic targets that have been loaded in these nanocarriers, and also the diversity in their application, which open really doors for new treatment and new diseases in human. Next, please. Next. So this research represents our lab focus, where we focus on nanomedicine, and we try to deliver and explore different areas within nanomedicine, focusing on designing responsive nanocarriers, as I will describe later on theranostic, combining imaging and therapeutic agents, exploring novel materials, and make them also suitable for specific targeting and delivering biologics, and also the production using like microfluidics. Collectively within the nanomedicine area, we try to apply this like to certain type of cancer, targeting specifically the cancer cells or the tumor microenvironment that has played a key role in cancer progression, as well as introduced new treatment like photothermal therapy to treat cancer. Next. So liposomes is one of the key nanocarriers that we work in our group. The fact they have been used for many years, they are clinically approved and very stable and be suitable for a wide range of therapeutics. We utilize them as small version of our cells. They consist of phospholipids where they encapsulate hydrophilic and hydrophobic drugs. We see the lipid bilayer here that mimics our cell membrane, and they have huge versatility in terms of the composition and the size that could be manipulated based on the application. This is where I use the liposome as a platform for a lot of application, as I'll present later in the slides. Next, please. So these are some of the examples of liposome, and most of you may be aware of Doxil, which is a doxorubicin formulation that encapsulates cytotoxic drugs that have been used in cancer, but also liposome could be used for antifungal agents like ambisomes and a lot of other applications that have been on market for several decades. Next. And you can press maybe all the animations, so that's fine. So one of the more important limitation with like conventional treatment, it's really the drug once it's administered, it results in distribution within the whole body, reducing the effective local drug concentration. This is mainly a problem with cancer drugs because of their cytotoxicity. So encapsulating the doxorubicin, as I mentioned, with the Doxil in a liposomal formulation, in fact, aim to reduce the systemic toxicity and increase accumulation at cancer site, as we see here, utilizing the enhanced permeation and retention effect. So looking at the leaky vasculature in the tumor, the longer the particles stay in the blood, they can accumulate more, and that has been, in fact, the main driving force for Doxil licensing. It's to increase the safety of the drug, not to efficacy. So despite the higher increase at the tumor site, this increase did not really result in higher therapeutic efficacy in patient due to the high stability of the formulation. So the drug is not bioavailable to deliver these cytotoxic or to kill and interact with the cancer cells. And that was a major issue. So we are improving on accumulation, but in fact, we are not improving in bioavailability and efficacy. Next. So several approaches have been targeted in nanomedicine, and the main focus is to either looking at the outside side of the rim, as we see here, that to increase the accumulation at site of the site of interest, for example, using monoclonal antibodies or targeting ligand. So all of this will increase the accumulation, but still has not resolved the issue of bioavailability. Looking at the inner side of the circle here, we can see, in fact, now we have this controlled drug release where we adding external trigger or internal trigger to improve the drug release and increase the bioavailability, and some of the example, temperature, magnetic heating, or pH. So all these things really improve not only on getting the drug to the site of action, but increase the efficacy and the bioavailability of the drug. Next. And you can press maybe all the stuff, that's fine. So hyperthermia has been one of the key areas where it could be combined with the existing treatment in cancer therapy. So hyperthermia, it means increasing the temperature at the tissue, which has shown on its own to have direct cytotoxic effect. And it also could activate the immune system that collectively will result in better therapeutic outcome in patient. If used in combination with drugs, that also improve the efficacy and aim to reduce toxicity and the administration of cytotoxic drugs. So that also has been done, but in terms going back to the problem of bioavailability, if we can design a delivery system that's basically responsive to temperature, upon reaching to the heated tumor, this is where the drug will not only increase accumulation, but it will be released and increase the local drug concentration that will result in better therapeutic efficacy. So that was the theme for using hyperthermia in drug delivery with thermoresponsive system. Next, please. So hyperthermia, and you can animate it as well. So hyperthermia, in fact, has been used to treat cancer. No, I mean, if you just say, yeah. So hyperthermia has been used to increase the tumor temperature, and it's very important to realize there is a wide range once we talk about hyperthermia. Either we have mild hyperthermia, which is up to 41, 42 degree, and that result in minimal changes in the cell viability, but result in increase permeability and accumulation, but that's normally doesn't cause cell death. Going higher than 43, this is where we start to see irreversible changes based on the duration and result in DNA damage and protein coagulation. That could be irreversible, and that means the cells start dying. So by changing the temperature and duration, we'll have different responses, and that's very important to consider once we are designing our system. Next, please. So from now on, I'll try to take you through some of the examples and research we have been working on in different areas in cancer and hyperthermia. And the first example will be utilizing hyperthermia to trigger on-demand drug release from liposomes. Next. So going back to the liposomes that I mentioned at the start, liposomes, they are very versatile system. We're looking at the lipid bilayer as we see here. In fact, this 4 nanometer thickness lipid bilayer could be designed in a way that release the drug upon changing the temperature. Next, please. And you can animate slides, fine. So temperature is key. What do you mean by temperature sensitive liposome? Looking at the phospholipid lipid bilayer here, if we, for any phospholipid, we all know that they exhibit a phase transition temperature, which is basically the melting point. So above this phase transition temperature, the lipid bilayer will be more fluidic, which means it will release more the drug, while below the temperature, it will be more on the solid phase and it's more rigid. So considering the phase transition is key in designing the thermoresponsive system, and a lot of work has been done from the 70s where changing the temperature resulted in drug release, but that was not really successful and beneficial for patients for many reasons. First, it was the temperature very high that you cannot achieve in the patient. Secondly, the release was too slow, and considering the washout, it means we are not achieving high concentration in the tumor. Next. So this scenario changed in the beginning of 2000 when Needham and Durst developed this thermodex formulation by Celsion, where the breaking news was of this formulation that the incorporation of a lysolipid lowered the phase transition where the drug release achieved at 41, 42, which is achievable in the clinic, but also more interestingly, this burst release where in nanoseconds, basically the drug is released in the heated tumor and the high accumulation result in high uptake of the drug, the tumor site, resulting in basically killing the tumor. So that was really game-changing in looking in on-demand drug release and temperature-sensitive liposomes. Next. So to prove this, yeah, no, you can go next, that's fine. So to prove this is some of the early work that we did, where in fact preparing the temperature-sensitive liposome with doxorubicin and test their accumulation in vivo in combination with mild hyperthermia. We had demonstrated high accumulation, as we can see here, but that's not only that dox goes to the tumor, it was in fact released with the increase in fluorescent intensity, as we see here, and that resulted in the slowest tumor growth and longer survival compared to the traditional formulation or non-temperature sensitive liposome. So it was really effective in animal model, and a lot of clinical testing has been done with this formulation for different type of cancer. So the focus for us, oops, stopped sharing. Sorry, I don't see the slide.
Can you? Oh,
I don't see the slides anymore.
Oh, wait a sec. I'll try to. Okay. Try to. Okay. How about now? Still not able to see the slide, Prof. Or I already opened it. No? Still not able to see the slides?
Yeah, no, I see it now. Yeah, I see
Oh,
it.
okay,
Yeah.
okay.
Yeah. Okay, so yeah, going from the in vivo testing, we had to explore really and think, is it really the formulation that's very important for drug release, but also the cargo, what we load inside is something to consider. And if you go to the next slide, please. So in fact, what we showed in our work, and you can animate all slides, fine. So what we showed in this paper a few years back, that if we take doxorubicin, which is the drug used in thermodox, but load it in different ways. So we load the drug normally using a remote loading method where we change the buffer composition to drive the loading and encapsulation. And interestingly, with the cryo-EM, we noticed that with the sulfate, where we have this like coffee beans shape and docs arranged like nice crystals, while with the ADTA, we could see a different shape. And more interestingly, upon heating the sample, we can see acute different response. Complete release and quick release with the sulfate compared to the ADTA, as we still see some crystal here, and that correlated nicely with their quick release and uptake by tumor cells. So by changing the loading method and also the concentration, the amount of drug, putting more of the drug, that affected their in vivo release and their therapeutic efficacy. So if the formulation become less responsive and slower at release in response to hyperthermia, then that's really a problem, and that reduces the efficacy despite being temperature sensitive. So that was very important to know. And if you go next, please. So not only doxorubicin, but we tried to apply this to also some of the doxorubicin prodrugs, like this doxorubicin conjugated to 7 amino acid peptides that's cleaved by the prostate-specific antigen enzyme in prostate cancer. And the prodrug was developed by Merck to try to reduce the cardiotoxicity. So the enzyme is overexpressed only on prostate tissue. So the idea, only it gets to the prostate, it will be activated and exert the systemic toxicity. However, that was not the case if we look at the scenario in vivo, where they observed non-specific cleavage in the blood, and that increased the risk of toxicity. So we aim to put it in a temperature-sensitive formulation to protect and spare the heart, while only the heated tumor will release the drug to be activated. And in fact, the approach was successful, where we can see that lower accumulation in the heart with doxorubicin, in doxorubicin with the loaded formulation compared to the free prodrug, and the therapeutic response while only achieved if we apply hyperthermia to the tumor. So collectively, that utilizing the pleural blood circulation of liposome and the specific activation at tumor site resulted in higher efficacy, lower side effects that hopefully will result in better outcome in patients. Next. So the second example, I'll take you from this formulation where you can load the doxorubicin or a prodrug based on the application, but always the question we are asked, how do we apply hyperthermia? And in fact, in previous slide, it's very easy to apply it in the lab using a water bath. Even for animal study, if we stick the leg with the tumor in a water bath for 1 hour while the animal is sleeping, then basically that's doable. But it doesn't really translate if we move it towards like clinical translation. So adding certain elements in the formulation where we can trigger it with more advanced technology is really helpful. And this is what we try to see. Can we add certain elements to the formulation to enable like drug release and respond like to laser irradiation? Please, next. So in this case, we looked for different elements to incorporate in the formulation, and magnetic nanoparticles, in fact, they were on the top of our list. Magnetic nanoparticles, they are used for clinical imaging as an MRI agent, but also they have been shown interesting result in terms of magnetic heating, magnetic targeting, and a lot of application, despite also considering their safety profile. Recently, a lot of studies showed that magnetic nanoparticles in combination with photothermal agent, like with laser, result really in high heating and requiring very low concentration of the particle, which was a bonus. So we decided to take these particles And next, please. And incorporate it in our formulation, and you can animate, that's fine. So we can add them in our formulation by using very small hydrophobic iron oxide that will be incorporated in the lipid bilayer. And upon laser irradiation, as we can see here on the left, the temperature increases quickly to the phase transition that enable the quick drug release, like within 5 minutes, the drug is really released. On the top of that, we can utilize the magnetic property of the iron oxide to use it as T2 agent, where incorporating a lot of particles within liposome, in fact, had a significant improvement of their signal, which means we can track them as well within the animal or within the human body, which was a bonus to provide this on-demand drug release and image-guided drug delivery as well. Next, please. So in this paper, we managed to publish this and to using the same temperature sensitive formulation we did before with doxorubicin to add an additional element of iron oxide that helps to release the drug and provide therapeutic response using photothermal irradiation. But more importantly, we could see in the tumor that the signals, we have enhanced signals from the magnetoliposomes. If you look at the tumor here compared to the iron oxide in their free form, the fact they are packed within the small carrier, it magnifies their signals, which makes it even more sensitive to see this in vivo. So collectively, the magnetoliposomes could offer on-demand drug release beside the image-guided drug delivery, which is very important to offer like personalized treatment. Next. So finally, I'll talk more about what some of the recent work that we have developed to understand and develop biodegradable photothermal agents. So I have shown different examples where temperature is important to treat cancer or to trigger drug release, but very important to see what type of photothermal agent we could use that could be safe and translated to patient and considering biodegradability. Next. And you can animate slides, fine. So yeah, if you animate the two images. So looking in the literature about the photothermal agents, in fact, we have two types of photothermal agent. The one on the left, they represent the inorganic materials like iron oxide, gold nanoparticles, carbon-based material. They are excellent in terms of photothermal heating. Compared to the one on the right-hand side where we see the cyanide dyes, they are organic. They are not stable upon multiple irradiation, but they are biodegradable. So collectively, we are balancing the stability versus biodegradability, where you need something stable and biodegradable at the same time. Next, please. Next. So this is where we thought, okay, can nanomedicine help us with that and transfer some of the application, take it like repurposing or using existing drugs or therapeutic agent to establish and develop photothermal agents. And indocyanine green is the only dye that's approved by FDA and MEA. It has been approved for more than 60 years, but mainly as a fluorescent imaging agent. So it administered into patient from the 60s, but mainly it binds to human serum albumin that basically help us to visualize the blood vessels, and that could helps to measure the cardiac output, the living function, and different organs. But it's mainly used for imaging. That's the only indication for indocyanine green. Next. So clinically, it's used for imaging. However, a lot of preclinical research have shown that in combination with laser irradiation, we could use them as a photothermal agent where the temperature increase could treat cancer, or as a photodynamic agent where reactive oxygen species could be produced and kill cancer as well. However, they have limitations. So the short blood circulation is suitable for having them as an imaging agent, but however, as a therapeutic agent, we would like to prolong this. They also suffer from thermal degradation, photo instability, and aqueous instability, where nanomaterial and encapsulation could help a lot and overcome some of these issues. Next. So one interesting approach, mainly to overcome the photothermal instability, is to arrange them. So just taking these small molecules, imagine these every individual, they are ICG molecules. If we heat them up at 65 for 24 hours, they start to arrange themselves and stack on the top of each other like head to tail arrangement to give you what we call them J aggregates. So it's simply a process of aggregation controlled by temperature. And these aggregates, they exhibit different optical properties. So the peak in the light green represent the absorption peak for the ICG around 800 nanometer, while the IJ, it's around 900 nanometer, where this shift improve their photothermal stability. And in combination with the laser irradiation, they have higher stability to be used as a photodynamic agent or photothermal agent. As well, it can be used as a photoacoustic agent. So this is another way of imaging that's very useful for cancer treatment and imaging. Next, please. So they have a couple of papers published these J aggregate, but in fact limited information as using them as photothermal agents. So we went into like systematic investigation where we compare the J aggregates to the existing gold nanorods that known to be excellent and stable photothermal agent, and radiated them with 800 nanometer laser and looked at the change in temperature. And what we see here, different concentration of the J aggregate and the different cycles, it means each time we irradiate the sample for 3 minutes with laser, then we leave it to cool down, then we repeat the cycle again, and we want to see how stable this upon multiple irradiation. We can see, interestingly, at the highest concentration, we have good stability where each time we irradiate, we still maintain more than 60-degree temperature, which was comparable to the some concentration with the gold nanoparticles. Of course, gold heats better, but the fact these are biodegradable and safe, which means even if we give more to achieve the same increase in temperature, we are achieving the same outcome with higher safety and biodegradability. Next, please. So one thing to consider, we start to say, okay, if we prepare these material and we inject them, whether to the animal or later into the human, are they really stable under different biological conditions? And we start looking at the stability in like serum containing media or in the blood and collagen. And you can see over time, in fact, they dissociate quickly to the monomeric form again, where we ended up with the same problem of low photothermal instability. So in this case, we thought like, okay, this is where maybe despite having excellent property, we still need a delivery system to improve on their biological stability. Next. So we went back and we thought, okay, the first option we thought of was really liposomes. Can we use liposome to overcome some of these issues? And you can animate all slides. So one of the stuff that we thought of, not only maybe to get the preformed, if you just stick back, yeah. So not only to use the liposome to encapsulate the preformed J aggregate, but can we really even prepare the J aggregate within liposome? So we have in situ formation in a single step. And that was in fact doable. And what was more interesting, that with this approach that I will explain later, we managed to achieve the J aggregate as we see here, using very low concentration of the ICG and within short duration, like from instead of 24 hours, we go really within 2 hours to have them. And we relied heavily on the packing of the lipid bilayer to promote this aggregation. Next, please. So how you can click everything. So liposomes, basically, they are phospholipid, as we mentioned, lipid bilayer. So we were very careful on selecting the lipid bilayer, which bilayer helps this and promote aggregation. And we thought the rigid bilayer will promote it better than the fluidic one. So what we did here, we took all the lipid component, we mixed it with the ICG and prepared like a lipid film as normal, and we extrude to prepare our liposome. And that has been done with others, but no one reported the IJA before. The only additional step we did is to heat the sample after we form it for 30 minutes at 65, and that in fact helped promoting the IJA in the liposomes. Next. The IgA formation was really dependent on the lipid bilayer, so not all liposome would prefer forming IgA. If we look at the rigidity from right to left here, we are increasing the rigidity. So the DSPC formulation known to be rigid with melting point around 55. Liposomes, they had good size. In fact, the fluidic formulation loaded more of the ICG, but that was more on the monomeric form. So the ratio between the aggregates and the monomeric form, we see it's only increasing, and it's evident with the more rigid formulation. Liposomal morphology, in fact, was very similar, so we couldn't see anything, but the aggregates were there. And then we start exploring their biological activity and stability to see, okay, how do they compare to the conventional one? Next, please. So we looked first at the phospholipid composition, but was also important to look at the other components. We did a lot of investigation using cholesterol and polyethylene glycol lipid that's present in the formulation. And in fact, the higher cholesterol content and PIC content, they were key to form the aggregate and provide stability upon multiple heating, as we see here. But also they were responding well if we incorporate them into liposome and we load doxorubicin, similar to what we did previously with iron oxide. Upon laser irradiation, they heat up, the temperature increase is quick, and that result in quick release of doxorubicin as well. Next. So the stability, which was a main problem with the free J aggregate, we thought, okay, are the liposome able to protect the J aggregate from the biological environment? And in fact, up to 24 hours, you can see the peak was present, indicating very high stability compared to the monomeric form, which was very important and very interesting to see. So despite having a system that in one step we formed the aggregate, but they were also stable. Next. And the behavior in vivo was the key thing. So we wanted to see, do they really get to the tumor and provide the stability we observed in vitro? And looking at the fluorescent property of the ICG, as I mentioned before, it's a fluorescing agent. ICG is known to have very quick uptake in the liver because it binds to the albumin. So within 1 hour, you see high accumulation, and over time, it's completely cleared from the body. If we have the conventional IgA formed or the liposomal one, we can see, in fact, the liposomal was more fluorescent, which enables tracking it in vivo. We see utilizing the long blood circulation of liposome, good accumulation in the tumor, as we see here. And if we take like the tumors or irradiate them with laser, again, doing this multiple times, each time we irradiate for 5 minutes with laser, then we switch off, we see the tumor turning from blue to red, which means it's heated, and the temperature gets around 42, 45. Then it cools down again, and repeat this multiple times. We still maintain good photothermal heating, which means the stability was not only proved in vitro, but also in vivo. Next. And the key thing going, are they biodegradable? Do they clear up from the body? This was the key thing where we looked again upon injecting the free ICG, the conventional IgA or our liposomal formulation. We extracted the organs from 1 hour after administration up to 7 days. And interestingly, we can see the J aggregate, they have less liver accumulation compared to the ICG. Gradually over time, we start to see the clearance from the body with all formulation, but still it seems the IgA will have higher preferential accumulation in the kidney. So maybe it's the route of clearance for these for this formulation. But collectively, we have a strong evidence that we have biodegradation and clearance. We didn't see toxicity up to 7 days, but clearly it seems we need to have to wait longer to be able to clear it from the body. But the fact it's clearing over time and no cytotoxicity, that's a good sign and prove the point of developing highly photothermal, stable, and biodegradable agent. So just a conclusion in relation to our work with liposome and photothermal, we could demonstrate that by selecting the right lipid composition between the phospholipid and cholesterol other component, we managed to prepare in one step the J aggregate liposomes where they exhibit high photothermal stability, fluorescent stability for imaging. Also, they have good and favorable pharmacokinetics and tumor targeting capability. Efficient photothermal agent, which I didn't demonstrate here, but in fact, they were effective in killing tumor cells in 2D and 3D model. And more importantly, they confirm the IgA efficient clearance in vivo up to 7 days. So they represent a promising platform that could be used for different application, including like immunotherapy that has been growing these days. Next. So in this part, because we know that IgA, they have high potential, but due to the delicate nature and the limitation of the previous work, where only certain lipid could promote the J aggregates, we thought, okay, can we go back and maybe work with the preformed J aggregate and load them to widen their application in different type of carriers like liposomes, lipid nanocapsules with an oily core, or solid lipid nanoparticles. And in fact, we managed successfully to load the IgA in these different carriers. However, we need to be very careful in selecting certain components to make sure that we maintain high loading efficiency with high stability. So some components, if we are not selecting the right component, the aggregates will dissociate again and we lose the benefit. So we managed to publish this in a couple of papers, which demonstrated really the careful selection of the formulation could achieve high encapsulation efficiency with interesting property and different applications. Next. So a concluding slide just to show that basically nanomedicines and utilizing these nanocarriers, they could be used to provide a new innovation, redefine certain therapies, and converting and widening the application of existing treatments. By having a selective and targeted delivery and controlled release, we can have precision in terms of targeting the disease tissue, resulting in high accumulation and lower systemic side toxicity side effects. Nanocarriers, upon having the right design and knowing the barriers, they can overcome some of these barriers and provide advanced platform and solutions. And more importantly, the versatility and the multifunctionality of these systems by combining different imaging agents, for example, and therapeutics or different type of treatment, we could provide the next generation precision medicine tailored for each patient. Next. So finally, I would like to thank all the people in my team. They have been involved in generating some of the results I presented today, and for a lot of other students who are involved in other projects that I didn't have time to discuss, but also a huge thank to our funders that helped us to conduct the research, and thank you for listening. Happy to take any questions. Thank you.
Okay, thank you so much, Prof. Wafa, for the wonderful presentation. And we now arrive in Q&A session. So is there any audience here want to convey the questions to Prof. Wafa directly? I guess I saw the some raised hand icon during your presentation, so I thought it would be somebody going to ask. Yeah, maybe anyone? So maybe we are while waiting then I'll check on the chat box first, bro. Yeah. Already 2 questions coming up. So the first question from Mrs. Masito. Yeah. She is asking, what is the difference between temperature sensitive liposome, magnetoliposomes, and conventional liposomes in liposome components and formulations?
Sure. So basically, liposomes, they are very versatile. And the fact I did my PhD with liposome and I keep working with them because they give you huge flexibility. What you need to do is they are formed with a self-assembly process. The only thing you need just to select the right composition of lipids, and then with the lipid film, for example, a method, or with the new method like microfluidics, you can prepare them. Based on the application, so if you want to make them temperature sensitive, you have to make sure that you consider the lipid you include in the formulation in terms of percentage, but more importantly, the melting point we mentioned earlier on. So each lipid will have a melting point. So for example, TPPC has a melting point around 42, DSPC around 55. So if you want something to exist in the gel phase above the phase transition, you need to work with specific temperature. Considering the body temperature, which is 37, to prepare temperature sensitive, you need something to be gel in the gel phase, which is more rigid above 37 and more fluid below 37. And in this case, it will be rigid below 37 and fluid above 37. So DPPC has been interesting. DSPC could be used, but then heating it up and releasing in the body, that's a challenge because you can do this in the lab, but not in the human. So adding a phase transition that's with suitable temperature to trigger release, that's what's important for having temperature sensitive liposome. That's a pro term, which means that temperature sensitive they are not new. In fact, they were started in the 70s, but they didn't have a huge success first because people were using more high phase transition and a lot of lipid that required high temperature to trigger the release. And secondly, the temperature was too high that could not really achieve. You cannot heat the body temperature in patients like 245. So everything stopped till we went back to try, can we achieve this switch at lower temperature? And in fact, the DPPC was still the main player, but adding a surfactant like lysolipid, triggering this fast release was the key change. So traditional formulation, you can put any phospholipid, but then maybe you are working with lipids with different phase transition that they don't show conformation changes upon heating. So that's the difference. Maybe DOPC, it's fluidic already at room temperature. It has a melting point of -20. So by changing temperature, it wouldn't really affect its release a lot. The magnetoliposomes, they are magneto, it means they are iron oxide containing. They can be of any composition, but you are adding an additional component. In our work, in fact, they all has the same composition. So the DPPC, lysolipid, and PEG in the formulation was key, but then we start adding either the ICG in the J aggregate form for some study or iron oxide. The formulation for IgA was slightly different because we, as I mentioned, we wanted the rigidity so that they were not temperature sensitive, but they were responsive to temperature. And the idea not to trigger drug release there, the idea was to increase the temperature to kill the tumor. So they are slightly different approaches. Thank you.
Okay, thanks, Prof. Lopa. So I guess that will answer all the questions from Mrs. Masito. And then another question came from Mrs. Endangdia. Yeah. She'd like to ask about the magnetic liposomes. What kind of magnets are used and only used biodegradable polymers in the targeted delivery system, or do we need to add the ligands for that?
Okay. So the magnetic liposome, what we used, you can use different type of magnetic nanoparticles. They can really be prepared with different sizes, coating. It depends really what you want to use them for. For heating, the size is very important. So any change in the size, that result really in different heating profile. And in fact, that was one of the limitation where we tended to use them with the magnetic hyperthermia to increase drug release, but we were not successful. We need to have very high concentration of particles to use. But then using photothermal was very interesting because at very low concentration and regardless of their size, they can really heat up. So specifically for our project, we used very small nanoparticles, hydrophobic in nature, because we wanted to incorporate them in the lipid bilayer. But you will see huge work done in this area where basically iron oxide, whether small or large, could be loaded inside liposome or other types of derivatives. Regarding the poly Sorry, was I muted?
I guess so. I think, yeah, some technical problem
sorry.
just
Yeah.
raised. Yeah,
So-
sorry, Prof. Yeah.
So basically, going back to the question regarding targeting, the liposome or any particle could be directed to the site of action based on the EPR effect, which is the leaky vasculature that exists, say, in cancer, but it's not always in all other diseases. And in this case, you don't need to add the targeting ligand. If you want to make it more specific and you want to get them to the cancer cells, then targeting ligand helps because it improves the internalization. However, there are huge debates whether active targeting works in vivo or not. So you would see, I think, a split between why to bother with the targeting ligand if it relies on the passive accumulation. But it's possible to add targeting ligand if you want a specific type of cells to be targeted.
Thank you.
Sure. Yeah, thank you so much, Prof. And then another question. Lots of questions just came up, Professor. I think we'll address one by one. We still got some. Yeah.
Sure.
Okay. Another question from Mrs. Pramesti Prisma. So she'd like to ask whether it's possible to formulate natural compounds such as plant extract or isolated phytochemicals into topical delivery systems using temperature-sensitive liposomes or magnetoliposomes. And what are the main challenge to or limitations related to these formulations?
Well, liposomes, they can be really used with a wide range of therapeutic agents. So you can use them with small molecules, large molecules. The key thing is really you have to understand your therapeutic agent and then match it with the best system. It doesn't like work one size fits all. If you are not considering carefully first the nature of the therapeutic agent and if you want it to be released or not and the route of administration, then it becomes challenging. So working with natural product is possible based on their property. It could vary in terms of encapsulation and maybe stability. This has to be evaluated. But also liposomes in terms of topical delivery, in fact, they have been used for topical delivery, and a lot of cosmetic products, they are based on liposomes. It might be the easiest to deliver compared to systemic because then you have you still have barriers. The barriers are different from the skin, but to apply it locally and then make sure you apply the photothermal, I think it would work nicely because then you have minimal issue with penetration depth. But it is doable. It's hard to generalize the limitation. It depends if you want to have deep penetration and maybe if the liposome are not getting deep or if the release is not that fast, because in fact we noticed the release depends on the drug and the cargo as well. So the nanocarrier and the cargo. So sometimes we used, for example, going back to the formulation, the broad drug release was slightly lower compared to DOX release. So that gives us different profile, but everything requires optimization, but it's doable.
Okay. I guess, Prof, we need to address just one more question, because I got a notification from the committee that our
we delayed, so I appreciate that.
Yeah, sorry for that, Prof. And one more question, yeah. So the question from Iman Surya Pratama: how can we distinguish whether the improved therapeutic performance of IgA liposomes is mainly driven by better biological stability, enhanced tumor accumulation, or controlled IgA release at the target site?
Well, that's a very good question. In fact, it's very hard maybe to disentangle a different aspect. Collectively, we can see better improvement, but this is where we tried in our design always to look for the comparative controls. So the liposome, in fact, the liposome will increase accumulation because they have longer circulation. So that's one thing. The fact they are more stable, so they could result in better photothermal capacity. So maybe they go together because the fact you have higher accumulation and stability that improve the performance. But the main thing, we wanted to develop a formulation that has higher efficacy regardless. I mean, one system that could address two issues, that's, I think, a bonus, but it's hard to separate that. But liposomes, they can carry also ICG, where more or less they will have similar tumor accumulation. But the fact the IgA was better than the ICG, maybe I didn't show the results here, it comes not only from accumulation, but also from photothermal stability.
Thank you so much, Prof, for your great explanations. And I think because we're limited by the time, I will close the session then.
Sure. Okay. Thank you very much. And apologies
Thank you
for
so
the
much
delay.
for the wonderful insight in nanomedicine, Prof. I think it's one thing to read about this advancement in your papers, but it's quite another level to have them explained with such clarity and vision from our meeting today with you.
Thank you very much. Thank you for the invitation and sorry for the delays at the start.
Oh, maybe, Prof, one thing. Do you have any take-home message for our younger generation? That because the participants here actually half of them are students, still students in the bachelor grade, or probably also the master's grade.
So to have the take-home message for them to pursue career in the future.
Yeah, well, in fact, it's great opportunity to talk to all students. It would be nice maybe to be in person and talk to them directly and give the advice. But what I would say, it's very important to be curious. Research is about curiosity. A lot of project, in fact, the IJA project, it was not something planned. It was something we noticed, and then we took it further. So be open for any new opportunities. Be critical about things, and try always to have in mind like what's the application. So try to make it more focused research, focused approach where you would like to achieve something. It's good sometimes to understand the principle, because you cannot maybe formulate the perfect system without really knowing what's there. So be critical, be creative, have the courage always to explore things. That's really important for people addressing research. And just I would like to take opportunity. I think one of my students, Sari, is basically from Indonesia, and she's a PhD working with me. And I think she asked
Mrs.
questions.
Ansari, right? Yeah.
Yeah.
Ansari,
So
Mrs.
it's
Ansari,
great to be, and she's working, in fact, with natural products. So this is where you can see a lot of stuff coming from nature, but then you combine it with nanomedicine, and that helps to overcome. So her research is very interesting, and hopefully we have other opportunity to present this in one of your meetings.
Thank you so much, Prof. And curiosity is the key for us to go move
Exactly.
forward. Yeah. Okay.
Yeah.
Thank you so much once again, and to our participants also. Thank you for you all for your excellent questions and for making this virtual session so dynamic. We hope this insight shared during the morning and afternoon sessions would provide valuable lessons for sure for all of us before we continue with the rest of the conference program. Okay, thank you once again to our speaker,
very much.
Professor
Thank
Wafa
you.
Al Jamal. Yeah, it is really wonderful to have you here. So,
Thank you.
good day, everyone. Assalamualaikum warahmatullahi wabarakatuh. I'll return back the session to the MC. Yeah, thank you.
Thanks.
All right. Once again, thank you so much for Professor Wafa Al Jamal, PhD, for such an amazing presentation. Now, we would like to present a certificate of appreciation to our fourth speaker, Professor Wafa Al Jamal, PhD, for the insightful presentation and valuable contribution to this international conference. We will also like to kindly invite Dr. Apotheker Sri Haryanti, Magister Science, to join us on screen and deliver their certificate virtually.
On behalf of the head of institution of STIFAR Yayasan Farmasi Semarang, we would like to express our sincere appreciation to our distinguished keynote speaker, Professor Wafa Al Jamal, PhD, for the insightful and inspiring presentation delivered today. As a token of our gratitude, we are pleased to present this certificate of appreciation virtually. Thank you once again for your support and participation in second SINACON 2026.
Thank you very much. Thank you.
Ladies and gentlemen, it is time to take a break. We have 20 minutes for break time. Please return at 1:30 p.m. Western Indonesian time, as we will continue with the oral and poster presentation. Also, the success of this event would not have been possible without the contribution and support of our sponsors. So, I would like to say thank you for Meta Jaya Metiga,
Total Equipment,
Global Sarana Instrument,
PT Magenta,
Asosiasi Riset Ilmu Kesehatan Indonesia,
International Forum of Researchers and Lecturers,
Asosiasi Pengelola Jurnal Indonesia,
Lembaga Pengembangan Kinerja Dosen.
So, thank you so much for your valuable support for this program.
Before the break time, we would like to announce the division of oral and poster presentation group for SINACON 2026. The oral presentation session will be conducted in several breakout rooms, while the poster presentation session will remain in the main room. Therefore, participants may choose to stay in the main room to attend the poster presentation or join one of the breakout rooms for the oral presentation session. Also, there will be a nomination for Best Oral Presentation and Best Poster Presentation at the end of the session. So please make sure that you remain on the Zoom until the end of the conference. And to all the audience and presenters, please don't forget to fulfill the attendance link that's shared by the committee.
This is where the next